I believe we have bottom in view. We're at a depth of about 1,700 meters, or still about 20 meters or so above bottom. And just as a reminder to everyone who is just tuning in, this is the NOAA ship Okeanos Explorer. We're about 90 miles south of the equator, and we are diving in the Pacific Remote Islands Marine National Monument, specifically the Jarvis Island unit of that monument. And we're on a seamount that we just mapped on the ship last night, and we'll be diving between approximately 1,720 meters, and we'll see how shallow we get as we cross this or climb this ridge arm.
If we see too much cool stuff, we tend to slow down.
That's correct.
That's a good problem to have.
That is a good problem. The more interesting rocks in biology, the slower we go. The seamount has the proposed name Kahalewai, and I'm hoping later we will hear from Heidi Hirsch, who sent us some information on this, the naming process for these seamounts. And there's a rather interesting background essay that she wrote on this, and it has to do with the early Hawaiian colonists of the islands down here. Really interesting story. And you can read that essay on Ocean Explorer, all one word, oceanexplorer.noaa.gov, and go to this section of that website for the Okeanos Explorer, and then more specifically for this mission. And you can hear about, or you can read about that interesting story. We'll talk a little bit more about it later in the dive if we don't hear from Heidi.
Yeah, I hope she'll call in. It's a piece of history I certainly didn't know about, and probably lots of folks don't. So.
I agree, Del. It's not something I knew anything about.
Pilot's just mentioning there seems to be a bit of current on the bottom here. Dive site we were at yesterday seemed to be pretty current free, although we did see lots of evidence from the ripples in the sediments that there may have been stronger currents there at some time. Getting our first good view of the bottom here.
I can already see some corals, so that's another good sign. That's what happened the other day.
Yeah.
We got a shrimp here.
Is it? Is that Chris's drunken sailor shrimp there? He seemed to.
Did you see him twist onto his side?
I did. I didn't look like he was swimming too gracefully. Huh?
May not be fair to accuse him of swimming ingracefully when he's being pushed by ROV thrusters and the
That's true.
currents.
That's true.
I'm definitely liking what I see at the bottom so far. A lot more hard bottom, not much substrate, little sprinklings, excuse me, not much sediment.
Right.
Little sprinklings, and definitely some large coral colonies down there.
Yeah, so we'd expect to see some manganese crust in these rocks, just like we've been seeing on the other dives at these depths, certainly. And so that's probably what the black color here is on the surface. But yeah, a lot less sediment than we've really seen on either of our two previous dives here.
For people watching, just for some explanation of the methodology here, so we've been traveling down through the water column. We're using what's called a dual body system. Effectively two ROVs, a camera sled with an ROV below it. that are being lowered into the water from a cable from the ship. And as you can imagine, if you were throwing your fishing line into the water, your lure would sink if you just left it so that it's going more or less perpendicular. And so there's a bit of settling out. And so when we get into position, it takes a little bit of time to get the vehicles in position and get us all ready to do the dive. And boy, this is looking a lot like where we left off in the last dive, isn't it?
It is, yeah.
My
That's
goodness.
it's a big forest, I guess.
Wonderful. Yeah, these are definitely some of the same bamboo corals that we saw in the last dive. These very tall, sort of open, bushy-like colonies that are a little bit yellow tinted.
Right.
The white thing might be a little bit different at the bottom of the screen. Great. Pilot's watch lead. Terrific. So the large colonies that we're seeing here look like what we were seeing on the last dive. And so while we'd like to get a close-up, I'm more interested in that white fan-like one that you see almost directly in the center of the screen. And then we've introduced ourselves here in the back, so if you think that this is a good opportunity to introduce yourselves here in the front row, this would be a great time to do it.
Absolutely, yeah. So this is Dan Rogers as pilot. We've got Bobby Moore as nav, and Fernando Aragon as co-pilot, and Roland Bryan as video, and Emily Nero doing our highlight editing in the back. And we're all ROV engineers and videographers with the Global Foundation for Ocean Exploration.
Thank you, Dan. So we're going to get our first close-up here in just a moment. So if any of you were following along on the last dive, you know that we went through a bamboo coral forest, and we believe that these large colonies you're seeing are likely the same species we saw there, which is really interesting because we were some 500 meters deeper there.
Right, yeah.
We'll get a close-up and verify that that's the case. But I think our first close-up here is going to be maybe on that white colony.
It doesn't look too familiar from our last dive. I don't know if
No.
you saw that or not.
Copy that, pilot. That is definitely of interest. So if you didn't hear that on shore, we have a strong current moving. I'm sorry, Dan, you said north to south. Very good. So it's essentially north to south. So that is, let's see, we're on a west-facing ridge, so it's kind of pushing you into the ridge a little bit. Is that right? Lateral, okay. Right. So it's coming on the port beam, basically. Okay. In fact, pilot, it was the one next to it we'd seen before. This is the new one. No, this is good. No, this is the one I want. Thank you. You were ahead of me. So this is another bamboo coral, much like the one next to it, which is a type of Octa coral. And you can see the white skeleton will be interrupted every now and again by black bands or stripes. And one of the things that we look on, you can actually see it quite nicely here near the base, is whether the branches that are arising from the skeleton come from those black bands or whether the branch arises from the white skeleton in between those branch bands. And we refer to the difference there as nodal branching if it comes from the black part, which is protein, or internodal branching. So this is a nodal brancher. And then the other thing I saw in that quick zoom was the smoothness, the apparent smoothness of the polyps, and it was kind of hard to see through. I'm going to suggest that this is in the genus Jasonisis. And you see that it's a really nice, flat, planar colony, kind of like a big fan, whereas all these others you see are kind of open, loose bushes
Right.
with the branches going in many different directions.
Yeah, and Scott, this is Tim for Associates. I just saw several crinoids on there, sort of yellowish to brown, and also on the seafloor I saw a long string of barnacles. Looked like short-stalked barnacles. Thought was interesting.
Thank you, Tim.
Yep.
We're just going to get a quick zoom in this little cavity here in the bottom. We can see a tail wagging. The kind of an eel-like fish. If Bruce or Astrid want to make any comments as we zoom in here.
I'm still waiting for the zoom.
Copy that. Yeah, it's under the overhang, and he's kind of on the far right of that.
Oh, yeah, I would have missed that. Oh, gonna give us a little view here, maybe? Oh.
Yeah, it's definitely a true eel, but we can't say what family it is at the moment. Unfortunately, we're looking at its back, and that doesn't give us enough information. It looks like it's got a fairly high dorsal fin, the fin on the back, if that's the orientation of the fish, unless it's turned upside down, which could mean that it's a conger eel, a congrid eel, not a conger.
Yep.
It's also got a, oh, here he comes. If we can zoom back in again, it looks like it was putting its head out, and that would be useful.
Yeah, I think you're a bit delayed, and it's already tucked back in, Bruce.
The tail looked a little bit like the morphology that we see on the arrow-tooth eels and the cutthroat eels, which have a lower dorsal fin. So I'm not sure which family this is, if it's a congrid or a synaphobranchid.
We'll see if we can get another bit of a look. There's a bit of a strong current in this area, so it might be hard to hold station. Okay, Bruce, we're getting another close-up. And now he's sticking his head out, so you'll get a good view of the head in just a moment. Looks like it is pretty prominent nasal protuberance.
My impression of this is that the dorsal and anal fins are about equal in height, and from the shape of the head, I think this is probably one of the arrow-tooth eels in the family Synaphobranchidae, subfamily Iliophinae. I don't know if Astrid has other thoughts. Oh, that's a very good view. It looks, yeah, I think it's one of the arrow-tooth eels.
I'm also seeing...
Astrid comments that we don't see white lateral line pores, the dots along the side of the body. I see them now. They're pale. This could be in the genus.
Now too, Bruce, I think we might be looking at Iliophis. What do you think?
Yeah, I agree with you. I don't know what species. I was trying to see if Ken Sulak is on the chat room. He's not. He's an expert in this group. But I think we have a good guess at the genus. Thanks,
I think
Astrid.
you can move on, pilot. Thank you. I noticed also 3 brittle stars in the bottom there. Doug, do you want to say anything about the geology that we're having a close look at here?
Yeah, you know, the only place we see any sediment is sort of on the backside of this ridge. That's consistent with this current sort of coming from north to south. So, and the way the ROV is transiting now, more or less kind of towards the ROV. So when we got to the backside of that, it looks like a little bit could pond in that depression. But most of what we're seeing looks like really heavily manganese crusted rocks. But my guess is these are basalts, these are lavas that have been crusted over.
I'm seeing a couple of interesting things here. One, we have a little bit of a drop-off on the left side here, which is really interesting. And this is one of the things we were discussing earlier about the change in the slope of the topography is often associated with accelerated currents. And so that's where you would expect to see lots of these suspension feeders. And sure enough, you can see them, some of them over the edge. As I'm looking at the map, I think we're settling pretty much right on the crest of this ridge, so we're not going to spend much time climbing up a wall. So it'll be interesting to see if on the flatter part to our right or starboard, if it's going to look like this. But I'm also seeing down below, I think I just noticed a Metallogorgia, and in the foreground an Iridogorgia. We'll get a look at that once the new pilot is settled in. Great, so if we can get a look just going off the lower left of the screen, there's a colony that is going to be, I think, new for the expedition. So it's tucked down a little bit. Yeah, I'm pretty certain that this is going to turn out to be what's known as Metallogorgia. That's a genus name. It has no common name. Sometimes we call it the pink parasol. And this is in the family Chrysogorgiidae. We saw several of those in the last dive. We referred to them as the golden corals because their skeleton is golden color. But with the Metallogorgias, their skeleton is typically a lot darker. It has this very interesting growth structure where you'll see it has a stem that's approximately 1 meter in length, and then at the top of that you'll see a lot of dense branching. And 100% of the time when we see this colony, we see an Ophiuroid brittle star, one of them, right in the center. So let's see if that prediction holds.
You're going to break your odds here. All right.
Yeah. And Tim Shank, I think, can back me up on that.
Yeah, you're backed up.
Thanks, Tim. And sure enough, there it is.
This is the third or fourth time where you've called your animal here, Scott.
That's right.
I'm beginning to think you're magic.
So one of the fascinating things about this is we've seen this species, Metallogorgia melanotrichos, all over the world in all the different oceans. And every individual we see always has one of these Ophiuroids. And it's the same species all the time. And we never see that species of brittle star on anything else, not even on the rock. And so that's a really tight association. In fact, maybe, Tim, you could talk about that. You're the one who studies these associates and their relationship with different corals.
Yeah, Scott. Well, I'm just looking at this one now, and it has an unusual, it's a typical positioning within the coral, but its arms are very active and hanging, dangling down. Normally, it's very tightly wound around the central axis and center of the colony.
I agree. It's very loose.
But yeah, this is it's very unusual, this sort of behavior it's doing here, which we often liken to sort of a feeding behavior.
Tim, another thing that's kind of different is the skeleton isn't quite straight. I noticed the lower part of it is missing its tissue, and these branches look a little bit more open. The polyps maybe even a little bit bigger than I'm used to seeing.
Yeah, I agree with that, Scott.
Interesting.
It's unusual.
Yeah.
Would this ever warrant a collection?
Well, that's a good question. If it was a typical Metallogorgia melanotrichos, I would say no, because we have so many of them and they're always the same. But there are some unusual aspects to this one, I will say.
Yeah, I haven't seen the central disc of the Ophiuroid yet, but the arms look consistent with an Ophiuchreus.
I wonder
which
if
is
this
the genus of the Ophiuroid that's typically, yeah.
Yeah, I wonder. I think that this the coral anyway is in a reproductive state. I think that's what we're seeing. The polyps are really swollen near the base, so I'll bet it's egg-laying season. Basically, we actually don't know if they have a season. They may produce eggs year-round, but these polyps look extra swollen.
Zoom on that, and I can confirm the arms do look Ophiuchreus-like, and so I would suggest that that would be what it would be.
Yeah.
Yeah. I've just never seen the arms do this. It's very loosely coiled.
Sure. And you would expect that they should be doing it at some point. I mean, the hypothesis is that the brittle star is doing something to protect the colony. I mean, there's this tight, a close relationship. They must be doing something for each other. And I noted again on the lower part of this colony, the tissue was gone, and there were all kinds of other stuff growing on it. So maybe this particular colony is under a little bit of threat.
Yeah, I agree with that. And also, the main axis has that zigzag morphology to the growth, and I haven't seen that before either in Metallogorgia.
Yep. And normally, so when they're growing, if we were to catch this at a younger stage, there would actually be little branchlets coming off that lower part of the stalk, and they're all dropped once it gets to its full size, and only the branches at the top remain. We can see a lot of different organisms that have settled on the lower part of the skeleton where the tissue is gone. You can see the difference in color between the light purple at the top and then the black at the bottom. So I see a couple of barnacles. That's those white things that you see. Those are sessile crustaceans. And looks like it's tucked down there in the at the base of the rock. That's great. Thank you. And as you're making your sweep up above, there was a coiling colony. You may remember the name Iridogorgia pilot. That's the next thing we'd like to have a look at. Tina Molitsova from Russia. She's one of the scientists, is an expert in various things, coral, black coral, and tube anemones, and she agrees that the polyps are quite huge on this particular Metallogorgia. Normally they look like these little teardrops almost sitting on top of the skeleton. I'm also seeing that red fan in there. That's probably a precious pink coral, one of the coralliids in the family Coralliidae, and we did collect a member of that family on the previous dive. There's quite a lot of diversity. Correct. Copy that, pilot. I'm wondering actually if that might be a black coral, that thing, because now I'm up here, I see it's more orange-brown in color. So we're getting another look here at this spiraling colony. This is yet a different genus of coral in that same family, Chrysogorgiidae, that has typically a golden or iridescent colored skeleton. And one of the things that is constant about all the species in this group is that they have these really tiny little polyps on tissue that is very, very thin. You'll always see their skeletons look somewhat delicate. Mm-hmm. Certainly compared to many of the other corals that we see. I think there's about 6 or 7 species in the genus Iridogorgia, and on the last dive we saw an Iridogorgia magnospiralis. I'm not 100% certain if this is magnospiralis or splendens. You can see a shrimp is living in the branches there.
Oh yeah?
Also not an uncommon observation. So here you can see the polyps just kind of sitting on one side of these side branches of the main stem. The main stem is the one that's coiling, and then they have these unbranched side branches. There's actually one genus that does have branched side branches. It's another thing that we use to identify them when we're looking at video. I don't think so. The shrimp is on the other side, so I think this is a good enough view for us. Thank you. And then again, behind it is this large bamboo coral. I'm quite certain that that's what we collected the other day. And we saw literally hundreds and hundreds, if not thousands, of these colonies during our dive.
Yeah, it really went on for the whole dive. It was amazing.
It was spectacular. Yeah, on the upper left, and if we haven't gone too far, just below this, there's something that's reddish-brown. We may have passed it, pilot. Yeah, I think I misidentified it as we were going by. And we have a request in the chat room to have a look at it.
It's a do-over here.
A do-over. You can't do too many of these. It's a little yeah, there it is. It just came into view in the center just below the lasers. Yeah, this operation is much better moving forward and up a slope because you can't really see behind you, and get a little bit dangerous. There's also a stuck crinoid over there. Okay, so I was right the first time, or I was closer the first time. It's not a black coral. This, I think, is a Paragon coral, which is also called a bubblegum coral. And I'll look for some confirmation from my fellow scientists on shore. And I can see there are a number of Polychaete worms on this colony. I see at least 4 of them. These are related to the earthworms that you find in your garden, but they have a lot of little, not exactly legs, but paddle-like elaborations of the side of the body that have a number of bristles sticking out of them that they can use to push off the bottom. They're very common in the deep sea, and they're very common associated with corals. No, that's good. Thank you. That's just what I needed. And now we can move on to that smaller red one that was up in the area we were in, if we still have time, unless we're making a move already. Great. And then tucked in under the rock, there's a sea star, asteroid sea star. That is not one I recognize from the expedition so far. That may be something new. It's in a little bit of a tough place to look at. Looks like it has some fairly prominent spines coming off of the arm. Thank you. So we're on our way to look at another coral that's just up ahead here. It's kind of red, very sparse, thin branches. And I suspect this may be what's called the genus Swiftia. Copy that, pilot, yes. I should say roger that, pilot. I see another one of those sea stars in the background, so that might be in a better position to look. Yeah, I agree, it has more arms than one we just looked at. Astrid Leitner notes that it looked like spider webs in that cave down there. And this is something that we see fairly often. Presumably it's mucus, and we're not entirely certain what's creating that mucus. So this is actually another colony of what we just saw. So this is actually very instructive. What you see here is that there's a lot of exposed skeleton, and that skeleton looked black, which means this cannot possibly be a Paragon because they don't have an internal skeleton made of protein. Instead, they have a more consolidated, cemented skeleton.
So we're talking about the part here along the main trunk.
Exactly. You see it changes from pink to black. So this must be some kind of plexaurid. And not at all one that I recognize. It's really interesting. So I'm sorry, I was just looking in the chat room to see if we had any other comments from folks about what this might be, because it kind of fooled me into thinking it was a bubblegum coral. But again, they wouldn't have the same kind of skeleton. Living on the branches of that coral are, let's see, a brittle star and a crinoid. The crinoid's the one that's got its arms all fanned out and is pointing away from us. Those are also called feather stars. They're both related. They're both different kinds of echinoderms, and both of those are 2 different classes related to the sea star that we saw earlier, which is a 3rd type of echinoderm. There's a nice polychaete on the substrate on the left side, about 9 o'clock. He's got a kind of a yellowish-orange head. Now he's near the top of the screen. And we see this one often swimming over the bottom. And here he is settled on the substrate. So you'll be able to see very clearly these structures called parapodia. These are the outgrowths of each segment of the worm. And they're kind of like paddles. And they have a lot of these bristles. And the more bristles they have, And the flatter those paddle-like structures, the easier it is for them to swim. So you'll see a range of variation in different kinds of polychaetes in the development of those paddles. And this one's going to have very well-developed ones.
Oh, yeah.
That sort of greenish yellow that you see is the head end. Asako Matsumoto from Japan, who's also an octocoral expert, agrees with me that this must be a plexaurid. Yeah, so very often you'll see these things moving through the water column in kind of an S shape, kind of shimmying. They're beating those paddles very quickly, and you can see how fuzzy they are. And they make very effective oars. Thank you, pilot. Let's have a look around. Okay, if we're more steady, sure. I see Amy Bako Taylor has joined us, and she has a lot of expertise in octocorals. And she agrees that the polyps really look like Paragorgia, and so this is kind of confusing to us. We're going to get a better look here, a more stable look, and see if we can figure out exactly what's going on here. So that's our maximum zoom, folks. And as our scientists on shore are looking at this, I'll just describe some of the things I'm seeing. So the crinoid, the feather star, you're looking at it from the back side, is on the upper left. Got the whitish arms, and you see some really small feet-like structures. Those are called cirri, that they use to hang on to the coral or the bottom, and it's extended its arms out in the water column to feed. There's a much smaller one almost right in the center of the screen. And again, otherwise I'm seeing tissue that's stripped in some parts, exposing this proteinaceous skeleton, and pink is the living tissue with the kind of yellowish polyps extended. Interesting. So we're going to guess what I said first, the genus Swiftia. And that's what I thought it was as we were zooming in. Just got fooled when we looked up. Okay, thank you, pilot. That's good.
So the exposed skeleton, that'd be some kind of predation then, Scott? Or?
Quite likely is predation. I'm not sure what other sort of things would be down here to scour it. If it was in an area maybe where there was more sediments constantly moving by.
Kind of abrade it then?
Exactly, yeah. We have a lot of questions about, you know, what came first, the chicken or the egg sort of thing, because, oh, is that a fish? No, that's a shrimp. The shrimp's still in the water column. Okay. There's a stalked crinoid. I'll just point that out. That's the yellow stalk that's kind of on its own to the left of the corals. So we saw feather star. And, oh, there's an interesting patch of pink here. This is going to be the anthomastus. So we were answering that question at the beginning of the dive that came in from Facebook about what was that mushroom coral. And I said it was anthomastus. This looks like a little field of recruits of anthomastus, I suspect. And purple. So the purple is going to be what's called a Stoloniferous octocoral. That means it's an octocoral that grows like a ribbon over the bottom instead of putting up a skeleton. But you'll still see the same polyp structure, you just won't see the skeleton itself. Really beautiful color.
Wow.
Yeah.
Yeah, it's quite different than what we've been seeing.
And it seems to be tracing right along the edge. That's a really nice view of the ribbon there. Just went off the screen to the top. There you go. And you can see that the polyps arise kind of periodically from that ribbon. And you can see some white cross-hatching in the body of the polyp. These are the microskeletal elements, the sclerites that I was referring to. So virtually all octocorals have sclerites, and they're a real key component to identifying the species that we're looking at. And so it's one of the reasons why it's kind of hard to identify from video. We have to really have that under a microscope to be able to get it right down to species. I'm seeing even some very small individual recruits. That's great. If we could have a look at the pink stuff as well that was just on this rock. A number of barnacles, all the white things that you're seeing there, they look like acorn barnacles to me. They're closed up. They don't have their feet extended right now, which is how they feed. Astrid Leitner says this is her favorite rock so far. It's really pretty. Yeah, so here, I think these are recent recruits of Anthomastus. Now, it's also possible, so you notice right down here on the right we have two individual polyps, and they're attached to the rock. And then just above it to the right, in the top right corner, there's 2 polyps, one smaller than the other. So this is a great example of asexual reproduction. You had a larva that settled, began to grow, it would develop 1 polyp, then that polyp buds and puts out a new one. And it'll continue to happen until you finally get a colony that could develop into these ones that are bigger than the D2. Now, if this is Anthomastus, it doesn't grow bigger than the D2, but there you go. And just to its upper left, I see the feet being extended from one of those barnacles. They're twisting a little bit, and they would kick those, they're basically legs. They would kick those through the water column to capture things. Beautiful view. We can zoom out. Shirley Pomponi is asking what the yellow stuff is. Shirley, I'm going to ask you, what's the yellow stuff? Oh, and there's a hermit crab walking among. This is really great. This means that there's been a whole lot of larvae that have settled in this location at relatively the same time, because they're all about the same size. And one of the questions that we have when we see these areas that has such a high density is it because a lot of recruits got there at one time and then they all started growing because there's so much food, or is it that they're there for a very long time period? So this could be very informative to us. So this is great because we're seeing a hermit crab that actually has a snail shell, a gastropod shell in its back, and so often we see them with zoanthids and anemones.
You saw an anemone the other day.
Exactly, and the idea is that, well, there's not enough snail shells to go around, so they're using these anemones, but here you go, there are some snails down here, folks. Great. Well, we're already getting a lot of diversity of stuff, that's for sure.
This is the spot right here.
This is the spot. We've gone, what, 5 meters? And a large urchin down to the lower right. You can see those very long spines. I think they call that diademid urchin. I'll let the experts on shore. And please, anyone on shore, interrupt my excited narrative here.
Scott, this is Chris at the UHECC, and it looks like a Spinodiadema is the urchin over there right underneath this gorgeous ledge. And then there looks like some type of a stock crinoid over there behind it. Is that correct? Is that what you're seeing, or is that one of the embellopathes?
No, that, I believe, is a stock crinoid. We passed it when we were just coming up, and we're just looking at it from a different angle now. Good call. So this is another echinoderm. So now we've seen 4 of the 5 classes of echinoderm. This is Echinoidea, a sea urchin. And we've seen the sea stars, the brittle stars, and the crinoids. The crinoid just behind is the stocked sea lily that Chris was just referring to. So that means all we have left to find is that, do you know, Del?
I don't know. Yeah.
Is sea cucumber.
I'm observing.
I think this.
Go ahead,
Sorry,
please.
Del. Sorry, Scott. I think this urchin's actually from the genus Plesiodiadema, which is the same family, which was the genus that Chris mentioned, Aspidodiademaceae. But I think it might actually be from the genus Plesiodiadema. Chris, what do you think?
I'm trying to remember which has the even rows of the spines going up and which are offset. I can't remember which is which, if I'm remembering it correctly at all.
So one thing I'll just point out there is it has these very long spines. The spines are movable. They're actually articulated against the round globular test. And then you could also see much more flexible were the tube feet that were extending from there. Thank you, pilot. We can move on. And there you can see that yellow stalk.
Wait, Scott, what's
Go ahead.
that to the left? There, it looks like there might be an encrusting sponge just to the left.
Okay, pilot, can we zoom back in? Under the overhang, there's a grayish blue patch on the rock, just in the shadow. And that was the voice of Shirley Pomponi at Harbor Branch ECC, the Exploration Command Center, and she is a sponge specialist, and her keen eyes detected this encrusting sponge on the rock. So Shirley, if you'd like to say anything more, we'll get it into view in just a second.
Yeah, okay, it's a demo sponge, and it's very thinly encrusting. I can't say anything more than that. I could probably, no, I'm not going to wager a guess on it, but it is a demo sponge, so it's quite different from the ones we've been seeing before that were hexactinellids. This is in a different class called the demo spongy.
Great, thanks, Shirley. And boy, if we think it's hard to sample some of these corals, imagine how hard it is to sample something that's growing flat like that on a rock.
Have to get our putty knife out, I think,
That's
for that
exactly
one.
right. Oh, I think I see another urchin tucked under there as well after we look at the spine, a different kind of, after we look at the sponge, a different kind of urchin. There we go. Oh, that's a fantastic view. Really nicely lit, guys. That is wonderful.
Really beautiful. Really, really
spectacular.
beautiful. Thank you.
Oh.
We are at a depth of 1,694 meters. We are in the Jarvis Island unit of the Pacific Islands. Thank you. The Pacific Remote Islands Marine National Monument. Thank you, Emily. So just above here in the overhang, there you go.
Oh, yeah.
I think that's a different kind of urchin. Yeah, it's got shorter spines. The spines on curves. So Diva or Chris, what do you think about this one?
Okay, but first a little follow-up on that last one. I'm going to concur with Diva on it. I looked through the gallery, and it looks to me like Plesiodiadema has these offset, the way the spines come out of the test itself. So I think she's correct in the Spinodiadema, which I said is incorrect. This one here, I think, is in the genus Cenopodyna, and it's in the family Podinidae. That would be my best guess. And there's a number of these that are pretty deep species. There's also shallow members of this family as well. They have pretty stout spines, and they're really beautiful urchins. I don't know much about them other than that.
Great, thank you. Diva, do you have any follow-up? Let's see, there's a snail also just above that.
Chris did an awesome job, and I totally agree with his identification.
Thanks, Diva. All right, well, let's see if we can move anywhere.
You're going to be up all night writing the dive summary, Scott. Oh, gosh.
This is wonderful.
So yeah, we still haven't seen much sediment. We see some bare rock here, little patches every now and then.
Nia's asking an interesting question about the geological structure. How would it be classified? Because it looks like there's some possibly interesting weathering going on.
Yeah, maybe Deb, are you on shore there? What do you think about the overall structure that we're seeing here?
Oh wow, we're going to want a close up of this yellow one. This is different.
So yeah, Deb, Nia was just asking about the kind of texture and structure of the rock here. You have any thoughts on what we're looking at?
Well, I'll be interested to hear what you have to say, but for me, I think that this is so heavily encrusted with manganese that I don't know that we're really seeing very much primary structure at this time. You're seeing a lot of the manganese crust that's sort of overflowing or draping over the original rocks.
Wow, so if you want to turn over to camera 2 to get a sense of how big this bamboo coral is that we're looking at, this is just unbelievable. Unbelievable.
It's 1 kc or 2 kc she's trying to measure behind us here.
This is a lot of Casey's. This is even a lot of Dells. You guys should meet Dell and Mike. This is spectacular. I think this is maybe different from what we saw yesterday. We have seen these yellow corals like this up around Hawaii, but this is not the yellow tint that we were seeing yesterday. It's still a bamboo coral. So when we get into a nice position, we'll get a close-up and see a couple of things. So everybody on shore, we're going to be looking to see how it branches. Are the branches coming out from the nodes, that's the dark parts, the protein part, or is it coming out from the white part, which is called an internode, and that's made of calcium carbonate. And it's one of the things that's so different about the bamboo corals. We were just looking at that Swiftia earlier with that red colony,
right,
and you saw that exposed black. So all of that black was the protein-based skeleton. It doesn't have all that calcium carbonate. Bamboo corals are special because they're more calcium carbonate than they are protein. The protein has been reduced to just those little nodes, and the thought is that it makes it flexible. The other thing we'll be looking for are spines that are associated with the polyps. And we're seeing a nice decapod shrimp here in the foreground using what are called its pleopods or its swimmerets. Those are the legs that are on the abdomen. It uses those to paddle around. Wow, this is really yellow, isn't it? I can see this being used as a photograph to go on my blog on color and Octa corals. So we're studying closely here The tissue is, oh, there we go. So I can see in the background, I at least saw a node to confirm that this is indeed a bamboo coral. One of the things I am not seeing are any strong needle-like spines sticking in between the tentacles. That's something that's important for us to classify with. I don't see that. These polyps are much thicker, more robust, and more dense than the colony that we collected the other day. So it is definitely a different species. Now here's a branch point, and it does appear to be branching at the node. Great.
Off of the black part there.
Off of the black
I
part,
see.
exactly. So I think this is worthy of a collection if, Pilot, you believe that we're in a position to do so. This is a truly magnificent looking colony, and we'll just take a little snippet of one end so we can get an idea on what it is. And here's one of the associates. I'm surprised there's actually not more associates. You know,
some of the much smaller things have been covered. Yeah.
Yeah. Oh, there's a great view of the skeleton, so you can see where the tissue is removed, and you can see the white calcareous internodes and the black proteinaceous nodes. And what we're getting a close-up here of is an ophiuroid brittle star. This belongs to the family Ophiacanthidae, and one of the hallmarks of that family is those really long spines on the arm. And Tim, if you want to say anything more about the Ophiacanthid, please do.
Sorry, Scott. I'm just watching it, a bit mesmerized by the
It is
length
mesmerizing.
of the spines. They seem to be extremely long, even for an Ophiacantid. And I'm trying to get an image of the disc here, but
it's
we see these sort of more or less indiscriminately on corals. But when we do see them, we often see them on the same coral too. So they often are on bare patches and dead skeletons. So they're not obligate by any means. They're more facultative symbionts with the corals.
I'm also noting some really interesting texture on the thick branch behind here, and I don't think it's shadow. There's something going on.
Yeah.
It almost looks like
little ripples or veins
exactly.
or something.
So I don't know if there's something growing underneath the tissue or on it. That is not really a normal thing that we see in these octocorals. And there as well. Look at all that.
Yeah.
It's like it's sagging. It's old. I mean, that part of the skeleton is certainly
Corals
thick.
don't get wrinkly like we do?
Not to my knowledge. And the sheer size of this and the thickness of this, I mean, this must be hundreds of years old. It's my wager. We sure do. Thank you. Now, any place that's convenient to you, what would be ideal is if you could include at least one branch point in the collection. But beyond that, it's all up to you. Thank you.
Scott, I want to ask you about, this reminds me a bit of what we used to call a bramble bamboo in the Atlantic. Am I totally wrong with that, or just a bad memory, or is that
no,
familiar?
I don't. Yeah, Tim, that's not a bad observation. So the bramble is one that grows kind of like this disorganized thicket, and when we were seeing them in the New England seamounts, we always saw them small, so we didn't know really what sort of structure they took on later on. But if I can see through to the base of this, it all seems to be arising from a pretty common base and not from multiple points. So I don't believe it's the bramble, but it could possibly be related. And I'm just going to call up my database and have a look at the other yellow ones that we've seen up near the Hawaiian Islands. There's at least two that we see, one that is in the S1 clade, and that's referring to genetic studies of bamboo corals and organizing them into groups independent of names. They will be named eventually, but it's a way for us to tell something about the genetic diversity. And the one that we collected on the last dive that made up that dense forest, we're quite certain is also in that clade, but it wasn't the same as this. But there's also another yellow one, and I don't remember what group that is in, and I'm going to try to find out now.
Yeah, so just to remind everyone, if you look on camera 2, you can see the view down from Sirius. Kind of gives you a sense of the width or the cross dimension of this feature and the topography here compared to D2.
Yeah, do you know the downlooking dimensions of the D2? Just the frame?
Yeah, I don't. It must be at least 2.5 meters.
I was going to say, yeah, 2.5 meters.
Yeah.
Across dimensions of D2?
Down looking.
Yeah. We're checking the ROV team. We've stumped them.
Well, basically, how tall is it? 9 feet, so about almost 3 meters.
Interesting to note that I did see some bear skeleton in there, and it was uncolonized. Previous colonies that we've seen on this dive have been mostly colonized when they've been bear like that. Not all of them.
I agree with you, Tim. So I'm looking at a photograph in my database right now, and there are a number of similarities to what we're looking at in front of us. Overall yellow, except that that one was an internodal brancher. Branches were coming out just above the node, and it had a lot of associates on it. It came from 1,385 meters on Twin Banks in the northwest Hawaiian Ridge. I have to read back on my notes, seeing if Amy Becko Taylor from Florida State University has anything to say. She's done a lot of collection of Octa corals in the Pacific and the Hawaiian region. She did think it would be interesting to sample. I haven't seen any follow-up notes. That's correct, pilot. At least one branch point, please. All right, I will go on to OKEX. Can you hear me now? Okay. It's fairly brittle. It's not going to shatter, but it'll snap very easily.
All right.
Not sure if there's a, I guess there's a purple crinoid. Yeah, there's a purple crinoid on one of those arms down below. I thought maybe it was a fish swimming in there. Understood. It's a really large colony. It's another Iridogorgia colony off to the left. That's the coil spiraling one with the side branches. One of the things that's so interesting about this immense colony, we just said it before, is that there are so few other organisms on it. And it begs the question of whether there is some chemical defense that's going on inside this coral.
Yeah, so far we've just really seen that one brittle star there. That's right.
I did see on the other side there was a purple-colored crinoid feather star.
Okay.
I'm forgetting the name of that right now. I should know it. Chris Kelly, if you're out there, I know that you would know that purple-colored feather star that we see fairly routinely. Comatulid crinoid.
Sorry, Chris just stepped out.
Okay, thank you, Astrid. In fact, I see a second one on this side, just above the strip branches there. You see it?
Little purple.
Little purple thing, exactly.
Yep.
He's got his arms kind of closed up, so it almost looks like a little fish or a squid.
Right.
In fact, maybe it's opening up. There's a second Swiftia, and oh, there's a Pelagothuria sea cucumber drifting by. How about that? Wow, now we've had all 5 of the Echinoderm classes.
What do you call that?
That was handy.
Is there a name for when you see all 5?
It's a royal flush, I think.
Hat trick or something?
The royal Echinoderm flush. There's the name Sarametra. That's the genus name for that purple crinoid, Sarametra, S-A-R-A-M-E-T-R-A. There's another one of the urchins as the diva has just made a note. Plesiodiadema we saw earlier with the very long curving spines, and it's tucked down just in the cavity to the lower left of the bamboo coral. Kind of underneath that pink Swiftia coral colony that you see, the sea fan. I see from the base, it looks like there's 1, 2, 3, maybe 4 main stalks to this thing. And
It looks like they all come together, though.
right, exactly.
Yeah.
So that they all come together to one holdfast, or Tim Shank was earlier asking about the relationship between this and what we call the bramble corals that grow as a kind of a thicket. And it's possible, I suppose, that when they're young, there's a thicket, but then there are some branches that become dominant and take off. So it's entirely possible this could be related. Diva Amon has made a comment in the chat room that is one that I have said to myself repeatedly, Diva. I totally agree with you. And she says that it's hilarious that Pelagothuria, that's the genus name for that holothurian sea cucumber that we saw go drifting by. It looked like a big umbrella with a tube underneath it. That it was quite rare before the last Okeanos Explorer cruise. And now we've seen 3 or 4 during this cruise. In fact, on the midwater transect the other day, we saw at least 2, right, Amanda?
Yeah, definitely at least 2. And, I've been watching some of the descents and the ascents, and we've been passing them almost every time. We're seeing 1 or 2 of them
so
somewhere along the way. So I don't know if there's some certain conditions here that are just really good for them or what's going on. It's something I've never seen before myself.
Exactly, and they're referred to by the holothurian experts as the most unusual and rare sea cucumber, but they're in this environment that is so hard to sample, the midwater of the ocean, and now we seem to see them quite often.
Yeah.
Guess we weren't just doing enough sampling in the Central Pacific.
Yep. There are many who would agree with that.
Sorry, I turn off OKX and I go on to Pilot, and I guess you can't hear me on that that line. No problem.
Gonna need a bigger box.
Oh, I guess I have forgotten to say to those of you on shore, may the 4th be with you. Since we're using movie quotes here.
That's right. I'm sorry. Yeah, I went Jaws when it was Star Wars Day. Yeah.
So I understand from Bruce Mundy that Heidi Hirsch may now be in the ECC at on Ford Island at the Inouye Center, and so this, Heidi, would be a fantastic opportunity for you to give us a little bit of the history of, excuse me, Jarvis Island and how we got the names that we are using for these seamounts. So please do give us a history lesson.
Well, aloha. Thank you for this opportunity. Yes, there's a little piece of somewhat overlooked history called the Hui Panala'au project that took place from 1935 to 1942, where and that's the means, the Society of Colonists is one of the definitions. And there were about 130 Hawaiian men who were recruited by the United States government to help show our presence in the Pacific. And these Hawaiian men, most of them were from Kamehameha schools, boy students, or school for boy students or graduates. And so they occupied Howland, Baker, and Jarvis for 3 months at a time, 4 men or boys per island. And it was an effort to show our presence in the Pacific during the Japanese military advancement across the Pacific. And they at first put military folks out there, but they didn't work out so good. It was the Hawaiian men that could really stay there on the islands and live well, fish, and take care of the islands. And they recorded information for the Bissett Museum, like all kinds of species of plants and fish and corals and things, kind of capture some of the biology that was there. So anyway, this is just a small little tribute that we'd like to do to acknowledge 3 of these men who passed away during this occupation. And one of them, the seamount we're on today, his name is Carl Kahalewai, and the grand-nephew of him is listening, I think, on the line today. He's following along the dive. I spoke to him yesterday, and he's really excited about this. And not tomorrow, but the next day, then we have Joseph Hananui, and then the next day we have Richard Whaley. So those 2 seamounts will be attributed to them for their time and their service to the country. Oh yeah, so then actually, because of their efforts, President Roosevelt was president at the time. Finally, in 1936, the United States was able to take control of its islands. So it's a really nice. There's a nice movie that tells. And if you go onto the Okeanos website, we have a little background essay there with more information about this. So thank you to these 3 gentlemen who were part of the Hui Panala'au project. And I hope this helps everybody's help. Thank you for joining us and honoring these men by a small contribution of referring to the seamounts with their last names. There you go. Thank you.
Great. Thanks, Heidi. That was wonderful. And this is really a spectacular seamount we're on today. It's quite the sight, and we've got some great maps of it. So it's a wonderful story, and thanks for sharing that history.
Yeah, and I hope the spectacular scenes that we're seeing here add to that honor. Pilot, on the top of this ridge to the left of the coral, I saw what looked like a different kind of urchin. In fact, I think there were 2. We passed over one, and I think, yeah, it's just to the left of the high point. little purple blob. Yep. And I'll bet we're passing a different kind of bamboo coral there, but I don't think we're going to have any problem finding corals today. There's a Medusa jellyfish swimming through the foreground as we make our way over to this other pancake urchin, possibly. Here we go. Nice. Oh, terrific view. We're looking up into the bell. It's kind of interesting. I'm studying it and trying to be sure that this is indeed what I think it is, and I'm not even confident that it is a Medusa. That is some kind of jellyfish. I'm not sure I recognize that reddish structure in the center. Everything else about it says jellyfish.
So what would that reddish structure be potentially?
Well, exactly.
It's not a common sight.
Yeah, this.
I mean, that's where the stomach should be, but yeah, it does look Medusa.
That reddish structure does look a little bit like a salp gut.
Yeah,
they always have
exactly.
those. But
that's not the typical structure. It doesn't look salp, but that's not the typical structure of a Medusazoan gut either. There are some where the entire bell is pigmented, so you can't see the gut, but not like that.
Huh.
Yeah, it is possible that there's a parasite in there or something that got up into the bell that's making that color, and it's not actually part of the organism itself.
I was also trying to see like radial canals, so the stomach branches out into canals, and you often see gonads in those canals. I couldn't see that either. And that's
no, everything else is quite transparent. Yeah, that was unusual.
I'm also seeing, so we're on our way to this sea urchin, and I see in the background there's a very large sponge. So Chris and Shirley, I'm sure you're going to ask me to have a look at that later. We're at 1,685 meters on a ridge arm extending southwest from seamount in the Jarvis Island unit of the Pacific Remote Islands Marine National Monument. I apologize. And this seamount has the proposed name Kahalewai, Levi, excuse me. And you just heard from Heidi Hirsch telling us a little bit about the history of the colonization of these islands and some of the sacrifices that those colonists made. And this particular seamount is being named in honor of the memory of one of those colonists, Carl Kahalewai, who passed away when his appendix ruptured while he was living on the island. Okay, Diva or Chris, would you like to tell us about this urchin? Or anybody else who's on the line?
The best I can do with this one is Echinoderms, but that's just a family level. I don't really recognize it otherwise.
Thank you, Diva. That's farther than I was ready to go. So we're getting a good look, a good close-up look at the spines here. So we were looking at one the other day in Dell. It was in soft sediments, and the spines had these swellings at the end, which has been proposed as a way to be more stable in soft sediment and push yourself along. Here you can see the spines are pointed. They are articulated, so they can move. There's slight swellings. Yep. Yeah, exactly. Yep. And then the purplish structures you're seeing are the tube feet, much longer, much more flexible, and they have, they're almost kind of like little suction cups at the end. You see very mobile. The tube feet typically go up in these 5 bands around the test of the urchin, which is the shell of the urchin. And the way I like to describe urchins to my invertebrate zoology students is if you take a 5-armed sea star, take its arms and pull it up over its head and glue them all together, you now have a sea urchin. And so you can see those tube feet are going from underneath where the mouth is, curling up over the test. Nice view, thank you. I think the next thing we're going to want to see is a head, and to the left there will be a large sponge down a little bit lower and beyond. I'm seeing a second of the Metallogorgia colonies just off the screen to the left beyond this rock, so I'm not sure that we've seen any singletons in terms of the biology yet. So much stuff here. Yeah, it's pretty much pavement. Then I think we're looking in here at the top of the ridge crest, so we'll need to be at the edge probably if we're going to hope to see some loose rock. So Shirley, if you're there, I think we're going to get a zoom on this sponge in just a moment.
It's a hexactinellid sponge, looks like, stalked. I don't know what it is. I'm sure Chris and maybe others would know what that is. I know Chris isn't in the ECC right now.
I'm kind of cheating by going to the animal guide. Chris Kelly and others have put together an animal guide, which is available through the Ocean Explorer website. And on the sponge page under the family Euplectellidae, which is a family of glass sponges, there's something that looks a lot like this in the genus Bolosoma.
Yeah, I think those are the stalked Euplectellids, if I'm not mistaken. This is really pretty. It's this yellowish. Is your guide showing it yellow?
Yeah, it does have a yellow tint, Shirley, the one I'm looking at. It has no specific name. It's just called species B, so they weren't able to identify it, but it looks a lot like this.
Yeah. Also, I was noticing there are a lot of barnacles, not the stalked variety, the other kind, around coral we were looking at before, and they're yellowish, and I don't know if there's a yellow encrusting demosponge on those barnacles. I don't see them right now. Oh, yeah, Diva has pulled up the Bolosoma species B. Yeah, sure does look like it.
And in the foreground, I'm also looking as we're zooming in. Oh, actually, sorry, just the note that we just heard that there's a lot of brittle stars in this sponge. The photograph in the guide, the animal guide, also shows lots of brittle stars, Ophiacanthid brittle stars, the spiny-armed ones on that sponge. So that seems consistent as well. We're almost looking at the same photo.
Yeah, I'd say it's a match.
It's a match for sure. Yeah, so I'm looking at that coral in the foreground. You see the way it's kind of wiggling?
Yeah.
I think that's probably a bubblegum coral. Now I will go to the chat room to see how many scientists are disagreeing with me. Great.
Get some positive reinforcement here, maybe. It's possible.
Yeah, it's possible. So you can see lots and lots of these brittle stars. This is the Ophiuroidea, is the class in the Echinoderms that include the brittle stars, or also called snake stars. And again, you can see these fairly prominent spines on the arm. So I think these are also Ophiacanthids. We saw a more red colored version of that on some of the corals earlier. And then we see one crinoid, the feather star, tucked away there, just went off the top right of the screen.
Well, these seem smaller than what we were looking at on the corals. Is there?
Yeah, I think that these are just likely different species.
Yeah.
So this sponge, sponges are filter feeders, and so they are generating a current. Inside the matrix of the sponge that you can see are a number of cells that have what are referred to as collars. It's actually microvilli. It's extension of the cell membrane of the cell, and it forms kind of a collar, and there's one flagellum in the middle of that collar, and it's kind of like a little whip, and that whip beats back and forth, and that generates a small current, but there are millions of them inside the sponge, and so all together they're generating a current. They're pulling water in through these little, call them pipes or passageways from the outside. They're able to capture very small organic particles, take them into the cell and digest them, and then the water comes out through all these different spaces that you're seeing.
Yeah, some of these brittle stars are really dug down in there, you can see, yeah.
Yeah, so I wonder if they're taking advantage of that flow that the sponge is creating, or if it's something else. I'm not exactly sure.
And my guess is they're probably taking advantage of that flow coming out of those larger pores, too. And this one, this hexactinelle sponge is a little bit fleshier than ones that we've seen on previous dives.
Great. Thanks, Shirley.
That is a truly excellent question. So this particular brittle star is very mobile, and so I don't think it would have any problem climbing up the stalk onto the sponge, but I admit I've never actually seen that happen. The brittle stars do have a larval stage in which they would be swimming through the water column, so we couldn't reject the hypothesis that the larva settles on the sponge. It's a great place to live and spends its entire life here. That's a fantastic question, and if anybody on shore has some insight into that, I'd be really interested in hearing it. Certainly I could imagine, you know, earlier we were talking about the brittle star that was living on the Metallogorgia, and I indicated to Dell that that is a really tight relationship. You only ever see that species of brittle star in that one species of coral. So that must be a case where the larva is literally settling on the coral. I don't know that with the sponge. Before we leave the sponge, after we're done with the imaging, you're welcome. If we could get a look at the coral, the pink coral colony that's just in the front, it'll now be on the right side. Thank you. Shirley Pomponi is loving this view of the sponge. It's really nice imagery we're getting here with the slow pan around it.
Definitely more stars on the front face, if you can call it that, than the back side there. That's interesting. It may have to do with the flow, I guess.
Exactly.
Yeah.
There is going to be a directionality to the way the water is pulled in and where it's ejected. I'm ready. So it depends on the kind of pigment molecule, and that may depend on the diet. Most of these animals cannot synthesize the pigments that are causing the color, and so it's coming from their diet. And what I had been reading earlier this week is that the pigments can be free, but more often they're bound to something. So, for example, maybe they're bound to the siliceous spicules that make up the sponge, and that binding alters the way the pigments absorb light and reflect light, and the color that we see is what's reflected. Yeah. Yeah, what a great question. So you would need to be able to determine with certainty what species of sponge this is and do something on its genetics, and then look to see now if we got the same sponge in a different area and it was white or orange, is it the same species? And that would be able to answer this question about, is the environment controlling to some degree the color? Bobby, you're coming up with some really good questions today. You should write some proposals. You're welcome.
So are these barnacles that we're seeing here at the base again?
Yeah, these are a whole lot of gooseneck barnacles, and you can see all their legs extended. So barnacles are crustaceans. They are related to the shrimp that we've seen swimming through the water column, except these have decided to be a little bit more on the couch potato end of the spectrum of shrimp. And when they molt after settling as a larva, they shed and metamorphose so that they essentially glue their heads to the skeleton and kick their legs to the water column. And it's kind of embarrassing. I'm glad we did this zoom on this thing that I asserted was a bubblegum coral, because this is a bamboo coral, my specialty. It is nothing at all related to a bubblegum coral. But again, you can see the dark nodes in there and the branching at the nodes. And you notice the way all these polyps are, well, most of them are closed up. This is great. There's one branch on the back right where the polyps are extended.
Yeah.
You can see the tentacles. But all the others have contracted their tentacles. I'm not sure why they did that. It looked like that when we were approaching. And you can see they're kind of volcano-shaped. I'm not setting you up this time. They're volcano-shaped, and this is actually a characteristic of the genus Echinoderms, which was described by Les Watling and I. So this may be the genus Echinoderms, and if it is Echinoderms, it should be branching from the internode, and I can see down there that it is not. So much for that. I think it's time to turn it over to you, Del. Hi, Chris.
Hey, I just thought I'd shoot you a quick identification, at least I think an identification. Those brittle stars that you're seeing on the sponges,
yes,
they're distinctive in that they have that really sort of deep incision in the interradial regions. We've been seeing these for weeks, I think, and they're pretty widespread genus of Ophiocanthids in this area. These belong to the genus Ophioplinthaka, Ophio, Plinthaca. I was just conferring with Sabine Storr, who is the curator of Echinoderms at the Swedish Museum, and she was telling me this seems to be the sort of genus that we're seeing a lot in the pictures that I've been grabbing. I have no idea how many species we're looking at. They all seem to do slightly different things. If you remember from Santiago's dive, every time the ROV approached a sponge or a coral with these things living on them, they would jump off. And these seem completely unaffected by the approach of the D2. So, different species, or maybe, they're doing something different. I don't know. They're weird animals, and that's, of course, why we love them. But anyway, that's my little my 2 cents worth of taxonomic information for the day.
Did you hear the question earlier about whether how it is that the ophioids get up onto the sponge? Do you think these are climbing up there, or do you think there's any larval settlement on there?
Oh, well, the first caveat, of course, is we don't know anything, or we don't really know a lot about these particular animals. But if I had to guess, I would say that it would be the larval settlement explanation, because you'll note that there are just a ton of them on the on the substratum, on the sponge or whatever. I think, as you had outlined just earlier, that what happens is the larvae settle, and then they kind of send off a signal that alerts other larvae, hey, this is a good place to come down. And I think they just kind of completely sort of en masse eventually settle onto an ideal substrate. That seems to be the most reasonable explanation, because I've never seen a sort of en masse migration of brittle stars along the bottom of the seafloor. Very seldom, in fact, do you see them just sort of moving along the bottom on their own. So, to me, at least based on what I've seen, that would best fit, the evidence. It varies, of course, for different kinds of animals, but, brittle stars aren't, they're, it's possible they can move, and people have speculated that, that they just, it doesn't take them long to just be, it would take a long time, but they could, they have the time to kill, so to speak, so they could just do that. Sea stars are kind of like that as well. But, I'd say the larval explanation fits what we know best in terms of the number, the abundance, and so forth. But, and of course, the presence corresponds to the water flow, which, it's too bad in a way we couldn't sort of individually go down there like Aquaman and pick them off and put them in different places and see if they adjust themselves depending on where the water flow, was coming through the sponge itself. It'd be interesting, because it's usually a lot of these brittle stars have a feeding optima. It's not really fast. It can't be too fast and it can't be too slow. It has to be just right. So There's definitely something to the location. Anyway.
Great. No,
and
thanks
I
for
think
those
that
insights,
earlier
Chris.
sea star you saw under the crevice might be a genus called Lowfaster.
Lowfaster. Great. Thanks for that, Chris. Always interesting to talk
How's
to
that?
you. Yeah, that's
That was great.
Great throw-in.
Thanks a lot.
Talk to you later.
Have a good day. Bye-bye.
We are having a close-up look here at a sea anemone. And we see this morphology of sea anemone quite often. Every once in a while you see, like on the lower left there, it's got a branching tentacle, which is kind of interesting. Thank you. That's a good view. We can move on. We've seen this morph quite a bit. I should point out, we have not seen it on this expedition, but in our dives in the Pacific, we see that anemone quite often. So a lot of excitement there, and now it's looking a little bit more barren, interestingly.
Yeah.
And I think we're getting up more onto the main part of the crest of this ridge. So it'll be interesting to see how things change. I do see a couple of these whip-like bamboo corals. And I'm not sure if this is a whip in the center that's forked or if it's something else. Little bunny ears. We're getting the vehicles back in position here. And I noted as we were passing there was something that looked like 4 or 5 branches arising from a center point on the substrate. And there's a Crinoid coral that grows in kind of that morphology. And to my utter astonishment, we have not seen any Crinoids yet. And I wonder if that will be one.
Might have been it.
So I suspect this is now the 3rd, maybe even the 4th species of bamboo coral that we've seen on this dive today. Tim Shank put an observation in the chat room that around deep New Zealand, brittle stars move as armies, so big collections of them moving together. And Tim, I remember, and Les Watling, who's in the chat room, when we were on, I think it was the cornerized seamounts, we were watching what we referred to as paratroopers. There were a bunch of these brittle stars that were in a coral, and they all started abandoning. You could see them jumping one after another, then drifting down through the water column and settling. This may be a sponge that's in this cavity if we have a moment to zoom in. Yes, indeed. I have no idea what that is. I wonder if this is a young version of what we're looking at. Surely it's a sponge. And that's as far as I go. There's a hermit crab below the sponge and to its right.
Yeah. This is another hexactinellid. I don't know what it is. Sorry.
Yeah, me neither. I don't remember. Is Caulophacus in this group? Is that one of the possibilities of?
It could be. And and see those barnacles now too. Those are the ones I was thinking might have. I don't know if the shells are yellow because there's something encrusting on them or not. But anyway, go ahead and get a close up of this sponge. That's great. It's really interesting. I don't know if the white that we're seeing are just kind of like really like tracts of spicules, which they probably are. Really, really interesting.
Yeah, it's very interesting. Also interesting, and not meaning to detract from the sponge, but because we can see it, I see there's a snail in the background behind that sponge. And we don't see too many live snails down here on the ground. This kind of looks like the shell that the hermit crab was carrying
just
earlier.
what I was thinking, a future home for a hermit crab.
Exactly, and there's also a hermit crab down here, so that snail may want to get a move on. And I think that hermit crab also has a snail shell already. Huh. Yeah, but I can see the soft foot of that snail back there. That's interesting. I'm more interested in the snail, if you can get a look at it, than the hermit crab. Top left, correct. Thank you. That's not a silly question at all, and I am not certain. What I am familiar with in shallow water, they're using abandoned snails. So the shells are free, the snail is dead. Oh, look, and there's one of those polychaetes.
Oh, yeah.
So this is different from the one that we were looking at on the ground before, but it's doing that same sort of shimmy motion that I referred to, and you can see how it's using those long paddle-like extensions from the body to swim. but it's got all these serial tentacular-like structures at the head end, so it's definitely a different species of polychaete.
It seemed a little leaner than the other one, yeah.
Yep. Yeah, there's two of them.
Yep.
Huh. But those long parapodia are a good piece of evidence that these are mobile polychaetes. They're probably not living in tubes or anything like that. They would spend their lives moving over the bottom. So if we can get a quick look at the snail, that's fine. If not, let's move on to those whip-like things, because those are definitely something new. Thank you. Yep. It's in a little tucked away in a little bit of a tough location to see. Another shrimp. So you heard Shirley Pomponi talk about the sponge before. She was also referring to all these light yellowish brown barnacles, acorn barnacles that are growing, and we're wondering about the color. Is the color just the natural color of the plates, or is there something overgrowing them?
There seem to be some white ones right next to the yellow ones there, yeah.
Yep. Yeah, maybe at an earlier age, you know, the shells haven't stained or something.
Yeah, they're definitely smaller, yeah.
All right, let's see if we can get a bit of a better look at the snail. He's really tucked down in there, so it's tough, but you can definitely see that it's a live snail because it's got that soft foot underneath it that's extending, and it's pointed away from us, and I think I can maybe see either the proboscis, excuse me, the siphon is the word I'm looking for, which would extend from the left front side, and I use it kind of like a suction tube, and they can draw water in and kind of sniff out what's in the water and also pull the water across their gill. That's good. Thank you. Let's move on. So I wonder, oh, there we go. That explains it. There's sediment here. I'm like, how could these things possibly? Well, I was looking at these corals, and I said, those corals should be growing here. I don't get it, but they're sediments. So what do you think they are?
Now you're predicting what the sediments are. This is
that's pretty good. It's the corals that are doing it for us.
Oh, look at that.
We'll go to the fish first. Astrid, I believe you are on now.
Hi, so this is definitely a rat tail, so Macuridae. What exact kind of rat tail? I think these are a really tricky group, and I don't think I would be brave enough to venture a guess. I don't know that it's Kumba again, but kind of that's my go-to with these small, large-eyed, rounded-headed rat tails. But again, we got to get close-up shot of the head so that we can send it to some rat tail experts who know more about this group than I do. But it is definitely a rat tail. Very cool.
Thank you, Astrid. That was Astrid Leitner, a graduate student at the University of Hawaii, who's tuning in from the Exploration Command Center on the campus of the University of Hawaii. So if you're at the university, you should drop by the geophysics building and check out the wall of monitors and see this in really large view. So the other thing that we're looking at here are these stalks. And so I was remarking with some surprise that what are they doing there? These are actually sea pens, so it's another kind of octocoral, but we've seen some rock pens, if you recall, in the last couple of dives, and I said that rock pens are a kind of sea pen that are unusual because sea pens usually live in sediment. And so that's why I was surprised to see these. Okay. So we just saw those rippled sediments on the other side of this rock. At the base of this colony, they would have an inflated bulb that's stuck in the sediment, and they have one internal rod. It's hard. You can see it giving it that stiffness. And that's actually in a single polyp. So you've got this one single giant tall polyp with a rod inside it and a base at the bottom called a peduncle that sticks into the sediments. And then it has popping out of its own flesh all the other polyps. So it's a bit of a different structure, but it's still an octocoral. So yeah, if we could see the base.
Yeah, maybe we can get a zoom of the sediments when we get around here too. Would be great.
Whatever one's easiest for you to see. So there you go. We're already seeing they're poking down.
Yeah, they're really clustered here. We have this band of sediments. You can see the ripples here. Pilots have been reporting pretty strong currents as we've been down here. So wherever we do get some sediment piled up, it's not surprising we'd see these ripples.
The sea pens
So
are...
just a note
go ahead.
again on that fish, I think it's actually a Nezumia.
Nezumia.
I won't go further than that. I think it might be a Nezumia, genus Nezumia, but still macrid. Thanks, guys.
Thank you, Astrid. So Nezumia is a possible genus identification on the rat tail. So here you can just see on this one on the left, it kind of swollen just as it disappears in there.
Holding
so
it in there?
Correct. Yeah, it's kind of thick like a ball. We saw it kind of as a plunger on the one that was on the rock.
So I don't know. Is Gregor with us today again? You want to say anything about the sediments? I'm not.
No, but we're channeling him with a rippled sediment.
Okay.
It's a little pun there, a little geology pun.
Yeah,
channel. Sorry, go ahead.
So fairly coarse looking sediment. These look a lot like the sort of biogenic 4M things that we've been seeing at the other sites, to my eye at least. Yeah.
In the foreground there, you were just briefly focused on a crinoid. So one of the things that's interesting, I see there's a whole bunch of tubes. So those are probably polychaete tubes that are sticking out of the sediment, and there are many polychaetes, these are the worms, that can take sand grains and other foreign particles in their environment and glue them together to make a tube. So they would be buried for some period of time, and then they would stick their heads out and possibly filter feed, so they may have a bunch of fan-like tentacles, or kind of garden or groom or pick stuff out of the sediment. So that's one possibility.
So were these tubes buried at one point and the sediments moved away from the outside? They're actually building these up?
Actually here, there's one sticking out over on the...
Okay.
Can we zoom on the one that's sticking out to the far left at about 10 o'clock? Sticking out of the sediment, there's a whitish thing. That actually looks like an Octa coral up there. Further left, almost to the far left of the screen. Yep. Thank you. There are a lot of things, Del, besides polychaetes that will build tubes, but they can build them so they extend. It's not that it's winnowed away.
Right. Okay. Yeah, that's what I was just curious.
Yeah. This is an octocoral polyp, so this is not what's building those tubes. Huh. Almost like a little telescope, huh?
Yeah.
There's a little submarine down there. Well, I don't know if that's a juvenile sea pen, but that is what I'm going to guess because it's in the sediments. There's something called Umbellula as a genus, and typically it grows about a meter or so tall, and then it's got 5 or 6 of these very large polyps at the end of that, but it's got to start somewhere.
Right.
So maybe this is where it's starting. I don't think this is, there's one genus of octocoral that is not colonial. All the other 2,000 are colonial. There's one that's known from New Zealand. It's about this size, and it lives in soft sediment. It would be amazing if that's what it was. I'm kind of stalling here, waiting for some help from shore. Diva Amon thinks that's a good guess. So Diva, do you mean it's a good guess that it is growing Umbellula, or that it is Tyaroa? Tyaroa is the genus name for the solitary polyp. We've got a good view of that, so I think we can...
I was concurring with the Umbellula, Scott.
Umbellula, thank you, Diva. So maybe this is a juvenile stage, and it'll we'll come back in 50 years, and we'll see if it's grown into a tall one. But I just wanted to make one other point here. So one of the things that we're interested in is diversity, and you increase community diversity, the number of species, when you have different kinds of habitats. So here's a really good example of how the sediment allows these kinds of corals to grow. Yesterday we had the beautiful example of the bamboo corals on that rock out.
The hard rock, right?
You know? Here we've got it all.
I think we also have more kind of three-dimensional structure from the rocks themselves today. There seem to be lots more crevices than were on the ridge the other day.
I think you can continue. It's interesting. You can see the various Helipterus sea pens sticking up, an indication of where that sediment is pooling around here. Les Watling made the observation that they saw something like this up in Twin Banks in the northwest Hawaiian Ridge, that wherever there was a small depression and with sand, they saw these sea pens. So that's also an indication that either the larvae are actively seeking out the sediments in the channels, or that those that settled on the rock were out of luck.
Lucky fellows hit the sand. Yeah.
There's a different kind of crinoid right at the base of the screen, just went off the lower right, and it looked like one of the 5-armed type. It was brown. So, pilot, for future reference, if we were to see a solitary polyp like that in the sand again, do you think we could scoop it up with our shovel? All right. We'll see. We'll see what other people have to say. This Tyaroa is really unusual. Again, I said out of the couple of thousand, more than 2,000 known species of octocoral, it's the only one that's solitary. And it's only known from about 600 meters depth or so off the coast of New Zealand.
We're about 1683 here. Huh? So.
We're at, yeah, 16. So this is a lot deeper and quite significantly further west. East. It's the long way around if you go west.
Yeah, you can go either way.
Yeah, you can. Now, what happened to all of the hard substrate corals here?
That's a good question. There's not really a lot more sediment on the rocks. It's really down on these little low spots here. So
as I look at our location, when we settled, I said, oh, we didn't really settle as much on the wall as I was thinking, and yet those corals are telling me something different. Mm-hmm. We were on a significant edge. Oh, look at that. How'd you do that? They found another one. Well done. All right, so this is interesting that we're seeing another one for another reason, and that is, would we keep expecting to see solitary juvenile polyps like this? Or is it more likely that we're actually seeing a juvenile colony? And my computer is frozen. That's unfortunate. I'll have to ask Del to have a look in the chat room to see what people are saying. There was certainly one request for it, but I would like to get more of a consensus than that. And I think my computer's unhappy at the number of different things I have open.
If you're in favor of sampling here, let us know while we have our window.
Yep, standby pilot. We'll see what we're getting from shore.
Tim says he's fine with it. The folks want to.
Asako. Yeah, I think so. We've got 4, including myself. So yeah, pilot, if you think that this is something we could sample in a reasonable time, let's give it a try.
Yeah, if we can scoop some sediments, that's great too.
Yeah. And pilot, if it's helpful to you, if this is the species we think it is, it would be approximately 4 inches in length. So another 3 inches below the sediment. At maximum, I would say.
In this case, the sediment will be our volunteer, I guess, right? So,
yep.
That's right.
I think in her way, Shirley is interested in this collection.
Yeah, people are excited about the scoop, I think.
I'm going to make them go back and collect that encrusting sponge.
It's our first attempt with the scoop here in this expedition, so. What do you want to call us?
That is a lot smaller than I thought. Yeah, I think we should let it go. That's smaller than I thought was going to be in that scoop. Thanks for trying. Appreciate it. Do you want some sediment, Del?
Yeah, if we're here, we got probably. You want to?
If you,
yeah, pilot. You want to try to just get a scoop of sediment? Yeah.
We've got the tool deployed.
We got it out. Let's give it a try. See if it's within our wheelhouse here. That's, I guess, as much as you can get. Now, will it make it up to the surface? I guess we'll have to get another matter. Okay.
Don't put your sediment on my coral. If there's a lot of mucus, and there often is with bamboo corals, it could be a concern. So if you can avoid it, that's great. But I understand it's probably easier to go into the port side box. Right? I mean, right now we have something in port inner. Can I just clarify? Is it easier for you to go to starboard? Okay, that's what I thought. Just so try to avoid the port inner if you can. Thank you.
Geo. Yeah. Sample O2 Geo. That's great, guys. Thanks.
Yep, thank you, pilots. You're getting a lot of praise from shore on what was a difficult collection for that polyp. Everyone recognizes the difficulty.
We called it specimen O2 Geo. Although Scott will certainly look through there for anything bio.
Nia is asking about what the ripple marks tell us.
Yeah, so that it tells us that there are some strong currents here. We wouldn't expect that unless we have some decent bottom currents, and that's consistent with what the pilots have been mentioning. In fact, they were commenting earlier that the currents were coming from the north to the south, so sort of down this ridge, and we can actually see that's the way the ripples are sort of aligned. The crests are normal to that direction of flow, so that's consistent with what we've been hearing.
Sorry, I was just pointing because I believe that polyp just went drifting all the way across the screen from right to left, so maybe enjoying his newfound freedom.
That was O2. O2, yep.
All right, so we had an adventure with the shovel. That's the first we've deployed the shovel on this leg. We see it's very effective at scooping the sediments there. There are various comments. Everybody learned a little bit from that attempted collection of the polyp there. So we'll have a better idea if we see something like that in the future and how to position.
Yeah, when you get a new tool, it takes a few times to figure out how best to use it, how to deploy it.
Sure.
So.
Well, I think we were expecting that polyp to be longer, bigger, deeper. And so it would. I thought it would have not drifted quite so easily.
Yeah, it sort of blew away there in the currents as soon as we freed it up.
Exactly,
Right.
And a couple of people noted that if we try it again, we should position it so we're downstream and scoop upstream. Yep.
Yeah, we didn't have too many options. Yeah.
Yeah, that's exactly what I responded. The conditions of the rock were such that we weren't going to be able to sit on that side and scoop. Yep, exactly. You work with the conditions you're given. All right, so we're at 1,684 meters. The water temperature is 2.72 degrees. And we have been on bottom since shortly after 9:30. So we've been on bottom for about 2 hours now. And we started on the north side of a ridge that's trending from the southwest to the northeast to the top of this seamount that was just mapped last night, and the seamount has the proposed name Kahalewai. Kahalewai seamount. And we're on our way now basically along the crest of this ridge, and we'll be moving upslope.
Yeah, so the end of our track we planned here would take us up to about 1,500. The seamount itself has a summit height of about 950 meters. So we're sort of on the upper part of it. We're certainly not going to make it to the crest here.
And yeah, nor was that our intent.
Nor was it. Yep.
We were hoping to get up to about 1,500 meters, which I think is a local high on this ridge. Lots of those Polychaete sea pens in the sediment down below, to our left and our right and ahead. So you can see they're creating quite a meadow. Actually, there's a lot of them coming up straight ahead.
Yeah.
So I've seen areas like this. There's something white that's coming up on the far right. I don't know what it is yet. We're too far out. Perhaps a sponge. Far left, I'm sorry. How often am I going to do that? Oh, wait, I'm sorry. Just turn back to the right for a moment. The real right. What's this puffy-like thing right in the front center? Oh, that's a really large crinoid, I believe.
Wow.
Stock crinoid. Yeah, it's got very long arms, and they're probably branched. And that should be on the hard rock and not in the sediments. And this is I think this is a different one, definitely for today. There you go. So this is another echinoderm. So all these feather stars of crinoids that we've seen perched on the corals and on the sponges, well, this is a relative of theirs. It's also a crinoid, but it lives attached to the bottom on a stalk. So this would never be free swimming. And these have the common name sea lily instead of feather star.
What's that wrapped around the base there?
That's a brittle star.
Yep.
Yep. So there's two of them, another kind of echinoderm. In fact, I think, Del, you probably know more than me. I haven't really taken any paleontology courses, but crinoids are really common in the fossils.
Really common. This is the kind of crinoid I think of going back to my invertebrate paleontology classes. These are very common.
Thanks for that view. And so here you get a sense of, we refer to these as sea pen meadows. Nice meadow of these Polychaete sea pens.
Wow.
Yeah.
Yes, nice metaphor there, the meadow, yeah.
So there's definitely food in the water column because these are all suspension feeders. And we can see some particulates floating around. We refer to it as marine snow, the undigested and broken up remains that are floating from shallower water. And they still have energy content.
You can see the ripples here. They're all sort of aligned across the screen, so you can see the currents coming from the north to the south.
Hey, Scott, this is Chris Ma.
Hey, Chris, go ahead. You want to tell us about that crinoid?
Hey. Yeah, I just thought I would chime in because it was such an interesting and unusual animal. We didn't get too long of a view of it, but I think that was that new species of Crinoid that we identified on the last leg. I recognized it because it had that really unusual texture pattern on the stalk. That was one of the many new things that they had seen on images from I think it was Indonesian region that was documented in this Tunnicliffe paper that I had sent along a few months ago. But I think they collected one on the last leg, and it had a really distinctive pattern on the surface of the stalk and on the cup, on the calyx, on the sort of area below the big arm spread on top. So it was just interesting because, it's new diversity, and a lot of these stock crinoids are really important to the stock crinoid people. They almost every one of them is something that they look on really longingly because so many of them are either new or rarely seen. So
great.
In the little got a sea cucumber coming up.
Yep. Yeah. Stand by there, Chris.
Anyway, but.
Don't
we
hang
saw a little
up.
pentacrinid there for a minute earlier as well on that sandbank, a little tiny...
Yeah, the five-armed one?
Yeah, anyway, I'm done.
Well, no, don't be done. Don't go, because I think we might...
Thank you, and I'll chime in later if I see a starfish. Thank you.
Okay, thanks. So we're... Go ahead, pilot. Yeah, I think you did the right thing. It looked like a dead skeleton, so this was a better zoom. So this is a sea cucumber that looks like it's either somewhat inflated or it's just sort of raising itself above the bottom. So we were kind of chuckling earlier about the 5 different classes of Echinoderms. So we have got the Royal Echinoderm flush. We saw the pelagic one, which is very unusual. This is a much more typical place to find a sea cucumber, a holothurian. On the bottom, they're deposit feeders. You can see these lines of these little tentacle-like structures that are sticking up. These are the modified tube feet, as well as around the mouth. And so there should be 5 bands, just like there are in the sea stars and the brittle stars and so on. There are various modifications, so sometimes you don't see the 5 bands, but it's a good example of a soft-bodied holothurian sea cucumber related to all of the sea stars, crinoids, and so on. They do have ossicles in their skin, so they're calcareous little structures, and the shape of those structures is also used for taxonomy. And echinoderm means spiny skin. Thank you. That's good for us.
Great.
I think we can move on. Yeah, I don't see anything more here. What do you think? Is there any geology close-ups you need here?
No, I think
there's nothing loose.
Looks very similar. We've seen a lot more sediment. If you look over in camera 3, you can see the nav screens. Kind of the contours here are much wider. That tells us, of course, this is a flatter terrain. That's how that reads in the maps, of course. And so it looks like we're depositing a little bit more sediment here than we were on that steeper part coming up.
There's something that's stocked just going off screen, almost off screen to the left. It's about 10 o'clock. It's beyond the sediment ripples in the distance. It might be another one of these large stock crinoids that Chris Ma just called in to talk about, that he said was so interesting and relatively recent description of the species. In fact, I think there's 2 of them in a row. So what I'm being fascinated as I look at this dell is all of these sea pens in the soft sediment, but very few corals on the hard bottom.
They've sort of disappeared, haven't they?
They've sort of disappeared, and yet there's crinoids here. They're also, feeding out of the water column. That's a good view. There's also something that's reddish brown, almost dead center right now on the flat rock. I suspect that's going to be an Anthomastus, which is that sort of mushroom coral. And earlier in the dive, we came across that ledge that had several dozen of the very new recruits.
Right.
This looks like an older one. This is what it would grow up into.
It's kind of out here by his lonesome.
Yeah. The red one, please. Excellent. Yep. So this is Anthomastus. This would be what I would refer to as, quotes, an adult version of all those young recruits that we saw very soon after we got onto the bottom this morning, and there are a whole bunch of them. So it's interesting what you said. It's kind of lonely out here on its own. I'm not sure how the recruits got to this other place. I guess that means this isn't as good a place to feed as that ledge was. Thank you, Pilot. That's good for us. And maybe just a brief view of the backside of one of those yellow crinoids when we're done here. Either one is easier for you. Yep. So Chris had indicated that, maybe he was saying it was the 5-arm one we didn't get that long a look at. Maybe that's what it was. Pilot, just to let you know, if you are going to be tugged off, we'll be more interested in the white colony that is just at the far right of the screen than the crinoid, so I'll let you use the time accordingly. Thank you.
Yeah, Scott, can we get a snap zoom on the ophiuroid at the base of this crinoid?
Sure, Tim, we'll do that.
Thank you.
See also one of those urchins we've been seeing several times now. Was it a spitodiadema perhaps? Or plesiodiadema, I believe is the name that was settled on. Thank you, that's good. So we get a little bit of a view, just hold that. So one of the things that the taxonomists of the crinoids use is how the branches come together and the number of times that they fork and so on. So this is a really nice view that they can use. Also, but the number of ossicles, fantastic. Nice stuff. You can see the fuzziness there. That was all the tube feet of that crinoid. All right, I wanted to just get a look at this white one because I'm wondering if this is either correct in the center, lower center, thank you. If this is finally going to be a primnoid or if this is just a small bamboo coral. So primnoid, I use that term, it's short for Primnoidae. That's the name of a family of Octa corals that are characterized by the polyps almost like being in suits of armor. And that's another bamboo coral. Okay, oh, that one has an aplicophyron. You see on the right branch there, there's no tissue towards the base, and you see something yellow and coiled. That is actually a kind of snail, or related to a snail. It's a mollusk called an aplicophyron. There, oh, beautiful view. Oh, fantastic. You can see there was some darker color at the upper tip.
Yeah.
Yeah, that's where its nasty teeth are. It's radula.
So
it's got the scraping tongue, and it's eating
working
that
its
coral
way
polyp.
up there, huh?
Yeah, it's eating that polyp. They're specialized coral predators. Awesome. I mean, awesome for us to see. Not so awesome for that coral.
Yeah.
Thank you. We had thank you, Pilot. We can move on. We had a live interaction with some students on the Cook Islands yesterday.
Right.
And you recall one of the questions was, you know, what's one of the most exciting things I'd seen? And I said that it was an aplicophyron being able to see it feed on a coral because I teach, you know, invertebrate zoology, and some of the things we teach about we've never actually seen.
Right.
So having this opportunity to take this fantastic microscope into the deep sea and get these exquisite images and behaviors is so exciting. Go ahead, Nav, this watch lead. Yeah, so it's a good question, so this is a conjecture, and it's based on some of the observations I made when we were doing the dives in the Atlantic canyons, and I saw several individuals of these aplicopherans on the rock at the base of the colony. So my guess is that it would feed its way up to the top and then just drop down and then crawl up another branch. Whereas the sea stars that are coral feeders, I believe they work up one branch and then will move down a branch and back up another one. It's all a hypothesis, you know, we would have to see the observation. I can see in the lower center there's another Swiftia colony. We had an image of that earlier. This one has a yellow crinoid on it. Okay, I'll tell you in just a second, but if we can get a zoom on this colony in the center, we finally found a primnoid, I'm virtually certain. It's quite small. It's to 4 or 5 o'clock of the lasers. It's got some pink brittle star in its branches, and now it's just below the lasers. Nav, my hypothesis is that it would get to the end, and then it would release and drop and sink to the rock, and then crawl up again a new branch. Yep. Yep, this is definitely going to be a primnoid octocoral. In the deep sea, there are 3 octocoral families that have incredible amounts of diversity. They've clearly what we call radiated. You have species radiation, meaning there's an ancestor that lived in the deep sea, and from that ancestor, many, many species have evolved. Isididae is one, that's the bamboo corals. Okay. Chrysogorgiidae, that's the golden corals, and this is the third family, the Primnoidae. And so it's so surprising to me that we're finally taking this long to find one. And one of the things, if we are looking at this closely enough, you can kind of see that the polyps appear a little bit rough.
Right.
And that's because the sclerites, those calcitic microelements that are in their tissue, are formed like plates and scales, and they're overlapping, and so they're kind of like suits of armor. And it's the details in those scales that we use to identify it. And another thing we look at is you can see that the polyps here are coming off in rings. All the bases are close together, and so there's probably about 4 polyps that are all coming out in the ring together. And then finally, when they curl up, do they curl up or down? No, that's great. That was a beautiful view. And you also saw that it had an astroskematid ophiuroid in it. Astroscematidae is the name of a family of ophiuroids, and we see those very commonly on corals. I don't know if that's specific to that particular coral, but great view. So our first Primnoidae, I hope somebody was able to put that into C-Scribe. If you hear us mention C-Scribe, this is a tool that we're using among the scientists where we can make observations on the biology and the geology that we see, input them, and then they're combined with the physical data, our location, our depth, temperature, and they're all linked to the video. And so we can go back and review the video or ask to be taken to parts of the video that have certain observations. For example, show me all the Primnoidae that were seen at log during a dive. We're passing over another one of the urchins that we imaged before, this one in the sediment. Several more sea pens. This surface looks very rough to me, Del.
Yeah, this is a little bit different here, isn't it? So yeah, pilots, if you see a rock that is loose here, we'd still love to try to get one. Look, go ahead. This could be the kind of spot to check out. That one that might. Yeah, we can just give this one a nudge if we want, if we can. Yeah, I think it's all pretty crusted, so we're kind of might take what we can get if it's available. My guess is it's going to be stuck on there pretty well, but. Oh, okay. Is it too big to collect or something? Okay, let's move on, but just keep an eye out if you see something. Yeah, so if you're watching on shore, those little red lasers you're seeing are about 10 centimeters apart.
Hey, Del, this is Deb over at the Harper Brand GCC.
Hey, Deb.
I hate to say it, but this so far looks pretty concreted in place, pretty in place. I hope we can find something loose.
Yeah, it looks like that one was loose there, but it was a little too big to get in the basket. So let's try this one.
Yeah, that was kind of giant. It's really different than the other day when we thought everything was really cemented and then everything was loose. So it's really quite interesting to see the difference in the manganese crust morphologies.
Yeah, the crust on the rocks from our last dive are maybe about 7 or 8 millimeters thick. And so we are moving up north here, and as we move further north, we'd expect to see some thicker crusts. We're going to be moving eventually into what's called a prime crust zone, where we might get things that are, 10 or more centimeters thick. But yeah, everything we've seen so far looks fairly crusted in.
And so, Del, for that reason, it's going to be important that we get a big enough rock that we can try to get some fresh rock in the middle of all of that encrusted manganese, right?
That's right. So the bigger the rock, the better. Our first one, our eyes are a little too big for our box, though. But we'll see if we can get one of these. Yeah, it looks like that's attached pretty well there, huh? Oh. All right, he's ready. Thanks, guys.
Hey, Dell, I've got a 7-year-old boy named Finley here in the lab that would like to ask you a question.
Sure.
You got time?
Yeah, we got time.
Have you seen any smooth rocks yet?
Have I seen any what?
Smooth rocks.
Loose rocks? Not today.
Smooth rocks.
Oh, smooth rocks. Oh, yeah. Many of these rocks are smooth. So there's this kind of stuff that coats the rocks here. We call a crust, and that crust sort of precipitates or comes out of the water on these rocks. And so that's one thing that people are really interested in these crusts, because they have lots of rare metals in them, lots of things we might be able to use in various technologies. So these crusts are potentially a resource that people are interested in extracting and using in the future. So you see all these black surfaces here are in fact those crusts. Now, at one time, though,
maybe a follow-up question.
Yeah, go ahead.
Okay. Go ahead. Sorry. Go ahead, Dale. Sorry.
Yeah, as I would say, but underneath here, if we get beneath this crust, this would all have been lava at one point. This would have been a big volcano, and so the flows would have come down off of the summit and built up this big mountain undersea here. And since then, all this crust has come on top.
Have you seen any sharks yet?
Any sharks yet? We have not seen any sharks yet, but I'm keeping my eyes open. So we have sharks and we have giant squid are on the wish list.
So in terms of sharks, actually might be a dive where we're in a depth range where we could maybe see sharks. Sharks actually don't go very deep. They don't go down past around 2,000 meters normally. I don't think we've seen any records of them, which is actually kind of a mystery as to why they haven't really adapted to exploit deep sea.
Thank you.
Great. We're glad you're watching and listening.
Great. Thank you.
Thanks, Chris. Okay. Let's. Great.
So, Del, this is Deb again. Are we still looking for a rock to sample?
Yeah, we're gonna test this one here. I think it looks like it might. We're closing, or the way it is, it might be easier to hit with the craft arm as opposed.
Oh yeah.
What do you think?
See if it moves.
Oh, look at that. Beautiful. That'll be lovely if we can get that pilot.
Excellent.
So over to the right, we might be looking at a dead skeleton of maybe a piece of a Walteria hexactinellid sponge. Not sure, but that might be what it is.
Okay, maybe we'll see if we can do a zoom after we have this. Yeah, so we'll be interested in these rocks. Some group of people are interested in the crust, and so we'll be looking at these rocks to see what the chemistry of the crust is. Are there minerals that are more concentrated in crust in some areas than others, and some depth ranges than others? And then we hope that we can break into this, and inside we'll have enough fresh rock that we can actually analyze the chemistry of the lavas that would have erupted here and constrain the age. Oh, yeah. Anyway, Scott has stepped out for lunch. Anyone on shore want to comment on this little fella?
Hey, Dal, it's Diva. So we can definitely say this is an echinoid or theurton, and I'm currently trying to look up an ID. It looks quite small, so it's probably a juvenile.
I think it's a baby urchin.
Yeah, juvenile, but so it might be a little hard to ID.
Okay.
Because it wouldn't have all the features that it should have as an adult. But I will try and get back to you with something a little bit more concrete. But for now, echinoid.
All right. Well, hopefully it'll go in the box with us here. We're going to put this in the closed box. So yeah, if we can do that, just see if we can get that little guy in there to stay in there. If that's not too much trouble. It doesn't matter. Everything's going to be easiest. It won't. Yeah. Okay. All right. Still got him on there? Great. All right, I guess this will be O3 Geo here. All right. And now we can tell Scott when he gets back from lunch that I collected a biologic sample for him. That'll be great. Or we could just let him be surprised.
So while you're waiting to position that in, this show, I just wanted to point out that a lot of times on the undersides of these rocks are some really interesting encrusting organisms. And one of my colleagues from the Natural History Museum, Sergei Tawada, is very interested in some very, very tiny encrusting sponges that are like maybe 8 millimeters in diameter, so teeny tiny. So if you happen to see any of those, go ahead and scrape them off and, I guess, put them in the OGL buffer.
Yeah, Shirley, this is Amanda here. We aren't great at identifying tiny little sponges, but Scott and I have seen a couple things that we think might be sponges that we've been pulling off and preserving for you. So we will continue to search over the rocks and collect anything that looks like it might be sponge.
Great, thank you.
You're welcome.
So we're just going to do a quick zoom on this dead sponge here, Shirley. Oh, just see what's on here.
All kinds of stuff on there. Shirley, this looks like this looks to me very similar to what we collected the other day. Is that?
Yep, dead Walteria.
There's a ton of stuff on there.
Sulfuroid, sulfuroids. Lots of stuff on this dead Walteria skeleton.
Yeah, very cool.
Oh yeah. All right, Pilot, that should be good. We can move on. Thanks.
Like a sea star there in the middle. I'm not sure. Yeah. As long as I was in the room, I don't think we've seen this one yet today.
This looks a little different to my barely trained eye. So yeah. Let's just zoom in here.
I'll have the pelagic ecologist and geologist tell you all about this.
We'll probably discover something really great, Amanda. What do you think?
Hey guys, I'm sure Chris Ma is going to call in imminently.
Great.
But until he does, this is a slime star. It's from the genus Hymenaster, and we've seen quite a couple of these throughout this expedition so far.
Oh, cool.
Great. Thank you, Diva.
And someone else has pointed out that they're also called cushion stars as well.
Uh-huh. Interesting. Huh.
All right. I think that's good then. Thanks, pilot. We can move on up.
So, Del, this is Deb.
Hey, Deb. Go on.
Hey there. So it's really, this stuff is really interesting. We've been sort of going over these bumps where we see all these, like, what look like really possibly jumbled flows, although, again, covered in manganese. And then we're coming on these little local highs where there's a lot of smooth crust. It's interesting because this is not exactly what we saw earlier in the dive.
Yeah, that when we started, we were a little bit coming up the slope.
And we did. We sort of saw that what I think in any other place we would have seen, sort of exposed rocks. And then we kind of came up over onto that little ledge that was very, very smooth, completely manganese encrusted. And now we're in an area that just looks much more jumbled than it was before.
Yeah, agreed. We've got a lot more chance for rocks here, a lot more loose things than we saw earlier. Even though this is fairly flat here in the topography, so that's pretty interesting.
That is interesting. There's also a lot more things that kind of look like they could be collapse pits, but again, covered in manganese crust. So one of the questions I had asked earlier in our chat room was, does anybody see anything fresh? Is there anything that we could see that we could say, this is a relatively recent broken off flow? And I have seen nothing.
Yeah, nor have I.
So I've returned from lunch, and I understand you got a rock sample with a present, I understand.
That's right.
From Caitlin.
Well, she gave it away. We're going to let you
we're going to keep it a secret.
Oh, yeah. I just saw it in the event log, too, so you can blame them.
All right. Well, I'm going to skip out for lunch then, and Deb, feel free to chime in about the geology in my absence.
Can we get a close-up on this colony that's right in the center above the lasers? I believe this will be an earlier stage Metallogorgia. Maybe not. Might be a full-grown one. So we saw one earlier tucked on a rock, and we couldn't appreciate the full length of the main part of the axis, the skeleton. And it's very often we see that the upper growth limit for these is something like 1 meter to 1.5 meters, so between 3 and 4.5 feet tall. And then they have that array of branches right at the tip. And I had said earlier, 100% of the time we see these, there's a brittle star right in the center of those branches. And so, so there is one little set of branches about halfway up. So this is just getting out of its teen years, as I would say. And that doesn't, I don't know what the length of time actually is, but when they're really young, they have a whole lot of those little branches going up the side, and then those fell off. All right, there you go. It's got its brittle star tucked away in the center of the arms, one brittle star. And these are some of the earlier branchlets that I guess just haven't fallen off yet. Great, thank you. Yep, we sure did. Thanks. This is interesting because it's not as deep a purple as the ones we're used to seeing, but the structure is otherwise the same. This also, like the one we saw earlier, has relatively large polyps. This is kind of interesting. No, I don't either. They're virtually
Hey, Scott,
always
are we full
purple
zoom?
color.
Okay.
Yeah, we're still zoomed, Tim. I don't see the central disc yet, Tim. That's Tim Shank at Woods Hole Oceanographic Institution, and he's interested in the different kinds of animals that live on these corals. And so there's a...
You know, if you remember from the one we saw earlier in the dive, how the arms were outstretched in a sort of feeding mode, and this one you see is more tightly wound, more the classical position for this Ophiocreus ophiuroid on this Metallogorgia.
Yeah, good observation, Tim. And the other thing that's different about this Metallogorgia and the one we saw earlier is this doesn't have the swollen bases to the polyps, so it doesn't appear to be full with eggs. But Tim and Les Watling, if he's still in the chat room, and even Abby Lapointe, I see is at University of Hawaii, all used to seeing these. This doesn't have the blue-purple tint that we're used to seeing on Metallogorgia. Thank you, pilot. Another one of the Swiftia sea fans. That's the red planar sea fan that we just passed over. And yeah. Might be interesting to get a look if you can drop down. I realize you're going over that ledge. Yep. Understood. The one that looks like it's got a fork. Yep, that's what I suspect it is. If you followed along on any earlier dives, you may have heard me get interested many times when we see these bamboo whips that look like they should have the morphology of an unbranched whip, but they seem to have this aberrant branch. And the question is there something unusual that's happened that's caused it to branch in an irregular way, or is this just a normal mode of growth and we often see them before they have branched? The comparison we basically make is you can see some very small bamboo corals that have lots of branches, but here you see a very tall unbranched one and the branches develop much, much later in life. The question is why. So it's definitely a bamboo coral and the branch is somewhere tangled up in there. But we're getting a good look at the polyps, so that's nice. Can help with identification. The polyps are really densely packed in there together. And they do not have prominent needles, but I can see on the far left, about the third polyp from the top on the left side, you can see these white lines coming from between the tentacles and going down a little bit, not very far. But those are sclerites. They're needle-like sclerites. And what I'm viewing is that they do not extend out beyond the tentacles, so it's not like it has a ring of spines protecting it. Instead, it just has those short ones. So that's, among other things, one of the taxonomic keys we use. Thank you, pilot. We can move on. There's another Metallogorgia melanotrichos colony in the background. That belongs to the family Chrysogorgiidae. The bamboo coral belongs to the family Isididae. Both very common groups in the deep sea between about 600 meters depth and as deep as 2,500 meters depth. So I'm looking over at the map that we're using to do the navigation, and Del noted this earlier that we've come up excuse me, onto a relatively flat part of the crest, and which is why you're seeing more of these sediment pools and so on. But as we continue to go towards the northeast, we're going to start getting closer contour lines, which means the slope will be increasing and we'll be going uphill a little bit more. But for the time being, it's going to be relatively flat, kind of the way you see it here.
Scott, apologies if you've covered this already, but I'm curious why Metallogorgia are called that.
Oh, that's a really good question. And, I have tried to learn the etymology of that genus, and I have not been able to determine it. One thing I can say is that Metallogorgia is in the family Chrysogorgiidae, and that translates to golden corals. And I noted earlier that when you get a good look at these Chrysogorgiids, the skeleton does appear to have a gold color, or it reflects in that sort of gold. Metallogorgia, on the other hand, the skeleton is usually much darker, purple to almost black. And so I wonder if it's because it has more of a metallic appearance, and it gets that name.
Thank you.
Yep, it's a good question. I really love the etymology of a lot of these things.
Yeah, it's interesting.
I think we had, during our Reddit session, we had some questions on, who gets to name these things and why. And the naming is personal. The scientist who is doing the taxonomic description gets to choose the name. Watchley Pilot, can we get a close look at the yellow colony that's on the right of the lasers? Thank you. In the foreground. So yeah, you can name something based on almost anything.
Right.
Something about the appearance of the animal itself, or something about the location it came from, or the vessel it came from, or a loved one, or a pet. There's all kinds of crazy...
Anything but yourself, right?
Anything but yourself, exactly. You cannot name it after yourself. Sure, you could name it after a band. You could name it after a president, a famous person. Yep. It's not Metallogorgia. It's Metallogorgia. But
that
yes,
might be with the next one.
Good try. All right, so this is a plexaurid sea fan, and we'll have to get a bit of a closer look. At first I was going to say it's Paramercia, but now I don't think so because of the strong reflection of white that I'm getting. There's a group of the Octa corals that are referred to as the Holaxonians, and that name is referring to something about the structure of the skeleton. If you're taking a cross, if you could possibly, yeah, thanks. If you were to take a cross section through the skeleton, you would see basically chambers in that skeleton, and that would unite all of these different corals together. But if you've heard the term Gorgonian sea fan, that's basically referring to this kind of coral that we're seeing right here. And it's probably in the family Plexauridae. Masako Matsumoto is one of the scientists who's joining us in the chat room. She's from Japan, and she is an expert in octocorals, and she may have a bit more insight than I do into this particular one. So I'm seeing that the number of large octocorals that live on hard substrate are picking up again. We went through a patch there where it was mostly the sea pens in these sedimented channels, and we weren't seeing as many of the large bamboo corals and things, but they're picking up. All right, great. So we're a little bit more on the flatter face, so we can get in a little bit closer. You can see that there's a very prominent brittle star there. And Tim, if you would like to comment on the brittle star. So this could be, I'm not going to exclude that this is in the genus Paramercia. If you look at the very tip top on the right side, you see there's a polyp that's sticking straight up, and you can see those tentacles with its little tiny side branches, and then just below it you see what's called a collarette. It's a little bit out of focus right now, but there we go, it's in focus. And there's a, if you know the term chevron, refers to like the sergeant stripes. Basically you have sclerites, the microskeletal elements that are arranged in these rows that create that sort of pattern around the tentacles. So that's called a collarette, and that's something that's characteristic of Paramercia. Thank you, that's a really nice view. Another thing that's characteristic about Paramercia colonies, the name Paramercia, this is a genus, they very often have these astroschematid brittle stars associated with them. And you can see Tim was referring on the Metallogorgia we saw, the way they coil their arms up and wrap their arms up tightly around these colonies, and they're very often associated with them. And here we see a single individual. Asako Matsumoto is saying that she's only sticking with the family Plexauridae. She's not going to try to guess what it is any more than that. And the details of identification really require you to have a specimen under the microscope. But this is a really good view. Thank you. I'm ready when video's ready.
This is Tim. I just wanted to comment for a second. I was just checking the records when it first came onto this, and this is the same morph that was on the Crinoid that we imaged earlier about a half an hour ago.
Oh,
And,
really?
I
Oh,
was trying to see if I could
so you
pull
mean
it.
the Ophioid is the same Ophioid morph as was on the Crinoid?
That's correct.
Oh, interesting. Yeah, good
So I
observation
think
there,
that's
Jeff.
ninth. Yeah, that's I think it's our ninth Ophioid morph on this dive so far.
Great. Pilot was just noticing as we passed over on the right, there was another Metallogorgia colony. That's the one that's got like the purple poof. In fact, that one was purple, pink purple, unlike the white one we saw earlier. And it's really hard to see, but there were these really small branchlets coming off the main stem. And again, that's an indication to us about its relative stage of growth. When they're really young, they don't have all of those branches at the top. Instead, it's kind of like an antenna with very, very fine side branches. Then as they age, they start to develop the branches at the top. As they age more, they drop the branches along the stem and are just with the branches at the top. So here we have another whip-like bamboo coral, and then in the background I see two colonies of the very big yellow bamboo corals, and we collected a piece of those much earlier in the dive. And I think there's another one of the crinoids. We've seen several of these stalked crinoids. It's kind of yellow, gold, brown in appearance. And there's one just here in the foreground. And Dell is back, and we haven't gone very far, Dell. We haven't seen any real change in the topography. Okay. I just noted that there's a lot more of those large bamboo corals all of a sudden. So that's kind of interesting.
Kind of come back into that.
We've come back to that. The sea pens are gone. There's still some small channels of sediments, but there's a purple sea cucumber at the far left, just went off the bottom corner. This may be the same morph as we saw earlier. It's kind of hard to believe that purple sea cucumber is a very close relative of this stalked crinoid. And yet they share a lot of common features despite the fact that they have big differences because one is affixed to the surface and one is mobile. And also to this thing, on the right side at about 3 o'clock, you see that purple 5-armed crinoid. So that's a free-living version of the stalk thing that you see there.
Ah.
Yeah. Different species, of course. I think they're barnacles, Jeff. Yep, they're acorn barnacles. We've seen them quite a bit earlier. And I think I see like a one polyp recruit of an anthomastus down there. There may be, if we can just snap zoom on the crinoid that's about 3 o'clock just going off, I see some pink dots on it, and I'm betting that those are little predatory, yes, the stalk crinoid. I bet those are predatory snails on the branches. And I think I see that urchin is moving where the lasers are. It's taking a little walk, maybe. Sure enough, there's at least 3 of them. So these are snails, and they're snails that are specialized to feed on echinoderms, and we've been seeing them with some frequency on the crinoids.
Right.
They have a little proboscis, so the mouth is basically elongated on kind of like a little tube that they can insert into the soft tissue of this crinoid. And I don't know, they may release an enzyme to help liquefy it and then ingest that, or these actually all these echinoderms that have tube feet, they have basically an enclosed system of fluids, and basically the tube feet are hydraulically powered. So if you squeeze those enclosed fluids at one place, it's going to extend at another. That's how they extend the tube feet. So it's possible that that proboscis is sucking that fluid. I'm not sure exactly how that works. Thank you. Great view. I'm sure we've seen a few of them, but if it's moving, let's have a look. If it's just sitting there, we don't need to have a look. And I think it's moving. Yeah. And what would be interesting to see if it is doing it is that it is actually using the spines to move itself. And it can do that because the base of the spine is articulated to the shell and there's musculature there that it moves the spine back and forth.
Yep.
There we go.
A little bit.
Yep. It's kind of like walking on stilts. Just a lot of stilts. Good stuff.
I'd like to see him go down the slope, though. That's what I'm waiting for. So.
And I presume what they're doing is, they're sort of moving around, and then on the underside, you can't see it, but tucked underneath is where the mouth is. And urchins actually have very complex jaws. There's something called the Aristotle's lantern, which is a structure of all these ossicles that are fused together with really complex musculature, and the esophagus goes through it. And they can extend that out of the mouth so that you see these, I think it's like 3 to 5 pointed, call them teeth, and they scrape the substrate and feed on materials in the substrate. I wonder if those little white things are little tiny sponges. Surely, if you're around and seeing this, are we able to get any tighter a zoom? I'm looking at the little white finger-like projections.
Little tiny white, there's little white things
Yeah.
that are sticking up. I don't know, Scott.
Oh, they're sure small. They kind of, sort of have that feel, Shirley, but wow, they're too small really to see any detail. I'm loving the urchin though. Does it strike you as that's a lot of effort? It moves and then
it's got to rest.
Down. What else could it be? I mean, I suppose forearms is the only other thing I could think of, but they look more like sponge material than forearm to me, Shirley.
Yeah, boy, I can't tell unless, I know they're tiny.
Oh yeah, they're so small.
If you get closer, I wouldn't be able to tell.
Yeah.
They're all over there.
They're all over, yep. Thank you. Oh, and there's another one of these Plesiodiadema urchins down in that cavity, so there's 2 right nearby. Interesting. All right, let's see what else this ridge has to offer. We're at 1,659 meters depth, 2.7 degrees Celsius. I'm Scott France. I'm a deep sea biologist at the University of Louisiana at Lafayette. And I'm sitting here with...
I am Del Bonensteel. I'm a geologist from North Carolina State University in Raleigh.
Wow, this urchin this crinoid is being consumed by snails. Look at all those pink dots on it. And then after, we're going to want to look at all the white dots that are coming up on the rock. Thank you. No, this is something else.
Yeah.
Yeah, there's some snails, and I wonder if these are myxozoan polychaetes. They don't really look like it to me, but...
they don't look like much anything.
Indeed. Huh. Well, Chris Ma, if this is something that you recognize, feel free. So myxozoans are a really odd little kind of worm, but I don't really think that's what these are.
So some suggestion there might be some kind of sponge here, maybe?
Sponge, huh?
Yeah, that's what Astrid has a couple question marks after it, so
sure.
We'll clarify it with that. So.
All right, I think we got a good image of it anyway, so we can pass that around. Thank you. So I'm not sure if this is a whole lot of barnacles that are aligned, not exactly on the high points, but on either the high points or on the sides of this rock, or if these are going to be a bunch of sponges. Sure. That's absolutely fine. Looks like a Echinoderms arrowworm drifting across our screen. Yeah, there it goes, just darted up at the top. That's something that we usually see in the midwater trawls, not way down here in the bottom. Midwater transects, excuse me. Oh, is that what it was? That's not a Echinoderms. Oh, it might be a little squid.
Ah.
Amanda, you should have a look at this. Okay,
Ah.
sorry, over here.
Barnacles.
Yes,
yeah.
indeed. Yeah, these are
Yeah,
acorn barnacles. Right now they're closed up, so the shell is closed up, but when they're actively feeding, they would open you see that split right down the middle?
Yeah.
Open that up and kick the legs out. Thank you. So there's lots of barnacles in this area. Go ahead, Nev.
That's right. I mean, I'm game.
There's another Anthomastus coral we're going over, just left of the lasers, the red thing. We looked at that closely before, called mushroom corals. And very early in the dive, we came across a ledge that had dozens and dozens of young recruits of them. Yeah, so the pilot has seen a small sponge here. It looks like maybe a glass sponge. It's just to the left of the laser, so on shore you'll get a close look at it in just a moment. Yeah, this looks like a small version of one of the euplectellids that we saw earlier. Shirley, what do you think? Can you confirm or deny?
Yep, I agree. I agree, Scott. Nope, I agree with you.
Thank you. Yes, sir. This, I mean, this is still going to be helpful. But. I'm sorry, whoever just called in, we can't hear you very clearly, or sound very far away.
I haven't said anything yet. This is Chuck Messing.
Oh, hi, Chuck. Okay.
Hi.
Welcome, Chuck. Good to hear from you.
I just logged on. I've been in the Bahamas for a couple of days. I had a chance to do a sub dive, but didn't see anything really worthwhile. But I just said I wanted to say that close-up image of that new species of crinoid is extraordinary. And those little yellows look like might be Myzostome worms on it, but I couldn't quite tell. Thank you.
Thanks, Chris.
Chuck.
Chuck, sorry.
Thank you, Chuck.
Hi.
So that was Chuck Messing at Nova Southeastern University. So Chuck, thanks for calling in, and you may have heard him say here's another crinoid just covered in these things.
Yeah, still got those on there.
We'll have to figure that out. So actually, Chuck is one of the world's experts in crinoids. Maybe we can just get a quick zoom. Chuck, if you're still there, did he say?
I am.
Do you recognize what? We're going to zoom in here in a second, and there's these orange, almost like shelf fungi-like things that are on the arms of the crinoid. Does this mean anything to you?
When it comes on screen, I'll take a look.
Okay.
Do you recognize what we're going to zoom in here in a second? There's these orange, almost like shelf fungi-like things that are on the arms and arms. When it comes on screen, I'll take it.
Well, I see some, I guess they're eulemid or melanellid white gastropods. Those yellow things could be Myzostone worms.
So yeah, that's one of the things that I thought perhaps was a possibility. They just seemed like such an odd shape, but I don't know what else would be on here that would look like that. And we've seen plenty of...
Well, I've seen elongated ones before, but then, and these guys look like they're wrapped around the sinews.
Well, we're going to have to take a close look at that photo and video later, Chuck, and hopefully we can get to the bottom of that. We're moving along now.
Is this something we might want to get a sample of, these things, if we see it again?
If it's on the right crinoid, sure, yeah. Now, so Chuck, what did you say about the crinoid itself? Is that something you recognized?
It's new.
It is new?
We think it's a Crinocrinid, Chryno, and it's got an on-branch pattern that is unique.
And is it something that was collected on one of the previous expeditions?
Yes.
Okay,
but
thank
they
you.
did not get the entire. They only got the top of the stalk, so if you see a smaller one to grab, it wouldn't be a problem. That would be great.
Copy that. Thank you. We can move on, Jeff. We seem to be going through these different sort of fields.
Yeah, now we're in the barnacle field.
Yep. Chuck, if you could mute your line when you're not speaking, we're hearing feedback. Thank you.
Done.
Yeah, so we had lots of bamboo corals when we started, and then we saw a big patch of the pink Anthomastus, the purple Stoloniferous stuff, the fields of the sea pens. Here's the fields of the barnacles, I guess. And we're still crossing the saddle. Yep. Several whip-like bamboo corals. And this, I mean, this is a good example, so I'm not sure if it was just, I think it was just after you left. This is a sort of a typical morphology for one group of bamboo corals. They're unbranched, but we saw one that did seem to have one fork-like branches. In fact,
this one here,
so does this one, and so does the one on the left. You can see that it's forking at the top. So it's not so unusual, but we have seen examples where it looks like there's been an injury to the tissue, and during the repair, for whatever reason, a branch has come off. And so that's the question. Is it a normal branch or is it a response to an injury? Yep.
Yeah, no sediment up here, so we might.
Copy that, pilot. Thank you. Shirley Pomponi at Harbor Branch is suggesting that those little yellow things that we were looking up closely on the crinoid could be sponges. She said they had a porous-looking appearance. So that would be interesting. An interesting way to collect a sponge.
You know, but Scott, Tim Sankson thought they might be egg cases, so I think that's another very good candidate.
Yeah, egg cases I think is a really good candidate. The question is of what? Certainly.
This is just an unusual, it would be a very unusual substrate for sponges to be on. I've just never seen that before, but then we're seeing a lot of really odd things.
The beauty of exploration, odd things.
Perhaps, this is Chuck Messing again, perhaps the man to ask about those little yellow guys is Greg Rouse at Scripps, who's done an awful lot of work on the Myzostones and knows their diversity.
Absolutely, Chuck. And I mean, I think that's what makes the most sense. What I found unusual is that they seem to be laterally compressed and tall instead of dorsiventrally compressed.
Yeah, you have a much better view of it than I do. I thought they might be, some of them at least, looked like they were, could be flattened, elongated, flattened, but curled around on the pinnules, but I couldn't quite tell.
Right. I certainly saw that at one. Our pilot has detected a new observation, I think, for today, unless you guys, maybe you saw one
I think
while I was gone.
this is what we saw while you were gone, I believe.
Okay.
Was a cushion star? Or
slime
star, or cushion star?
star.
Yeah, we had a couple different common names.
And just a quick
like
look at the
slime.
yellow colony there. Also, I think it's just a very young version of these large, big yellow bamboo bushes that we've been seeing, but I've not seen any small ones.
Right.
So this
Yeah,
would be
here it
kinda
is. And this is an area where there are no big ones. We haven't seen one in a while now.
Very good point. Everything's sort of hunkered down to the to the substrate. Okay, yep, indeed. So that is that same yellow bamboo coral. So that's good. And did you guys get a good look at that slime star before?
We did. We got a nice
Okay.
zoom on it.
Nice zoom on it.
Yeah.
All right, then we can keep going.
I think that's something we haven't seen before. Is that
The slime star?
Yeah, until this dive, right?
Yep. Yes, I am. Thank you. It's kind of nice. Right next to the slime star in the foreground is a brittle star. So the slime star is the asteroidea, your typical sea stars. The brittle star is the ophiuroidea. They both have tube feet, but they move in very different ways. And you can see this one is moving each of its legs independently and kind of walks up above the surface. It's going to raise its central disc.
I see.
There
yeah.
you go. Whereas the sea star kind of glides, just the tube feet move, and the arms themselves don't move. Good view.
More barnacles.
Go ahead, Nav. Right, and I was following that discussion. So the objective is to basically stay up on the crest, but what I've seen is the edges look a little bit more interesting, and I suspect, Del, you can chime in, given the how cemented this all is, if we're going to find a loose rock, it's probably going to be at the edge, so closer to the edge would be better than the center. Just a reminder to anyone who's tuned in and is not familiar with the dives, the two red dots that you see moving over the substratum are lasers that are just positioned, I think it's above the camera, associated with the camera anyway, and they're 10 centimeters apart, so it gives you some scale of the environment.
There's another crinoid. It looks like it has more of those yellow things on it. Yeah.
This is really fresh, or, well, I don't know, maybe it's darker, this rock here.
Yeah.
Still manganese, I guess.
Still manganese crust, I think, but had a different texture there. That's for sure.
See a lone bamboo coral there in the background. Almost looks like there's a little tiny branchlet coming up from the base. So maybe that's one of the things that fools us. It is branching, but one of the branches is broken off. Huh. This almost looks like a big loose boulder cracked off at some point.
I won't ask if we can get that in the rock box. I know the answer. I'd love to have it.
All right.
That's true. We have to crane it up to the rock box, I think so.
Alan Murphy, who we were working with when we were down in the Cook Islands area, was saying that what was from the Mid-Atlantic Ridge, they had to get an awfully big rock to be able to get enough inside that wasn't altered that they could do their analysis on. So maybe that would be big enough.
Yeah, I think it might work. Yeah. But you know, things do move down slope here. You can see right where that one broke off. So. What's this on the left here? Yeah.
I think we're looking at the back side of a crinoid. It's like its head is dipped down. I wonder if it's broken. So one is facing up and the other is facing down, but it's two of the same thing.
Huh.
Interesting.
Strange.
Yeah. Yeah, very sad.
Yeah.
Look at that. So there we can see a couple more of those eulimid snails. And so if you look up, one of those things we're wondering about, it looks like there's an opening at one end of it. There we go. So that, I think, is going to rule out the sponge idea. Huh. Maybe this isn't the same thing. Oh, I wonder if that could be an apocophron.
This is Chuck Messing, and it looks like they do an umbrella thing. Crinoids do an umbrella thing like that when there's little current. So they may. It's unusual to see one tilted all the way over like that. This is a new species, and the one to the right is sort of a typical weak current umbrella posture. But this bent over one, that's not typical. But then this is a different kind of stalk.
So do you think the one on the left and the one on the right are different from each other, Chuck?
No, they're the same species.
Okay, thank you. So basically they would take that umbrella posture because there's not much current, so it's more particles falling straight down instead of coming at them horizontally. Wow. Yeah.
Correct.
Thanks, Chuck. We're getting a really good look at these, the other orange things that are on the crinoid, and you can clearly see there's an opening in them, which I guess doesn't rule out sponge if that's an osculum, but they're not moving at all. Yeah.
That brown one, the orange tube-like mouth, these could be, there's a snail on my screen now. Yeah, I see that. Some mysostomes make galls. And they're really parasitic, so it could be, that could be a response by the crinoid and then the mysostomes inside there. But we'd have to get Greg Rouse, his opinion on this one, because he's the past master of that.
That's the best explanation I've heard so far, Chuck.
Oh, definitely, it's a new crinoid species. Yeah, it was seen in Indonesia during the 2010 expedition. And they collected one on one of the earlier legs, but it didn't appear to have these little tubes or whatever they are.
Exquisite. Interesting. So one of the things Chuck Messing was saying earlier is that if we saw a smaller one of these, it would be nice to try to get the whole thing. The collection that was previously made on one of the earlier OKEX expeditions only managed to get the very top of it. So they don't have all the characteristics. It's pretty big. But I don't know if it's necessarily the holdfast itself.
That is remarkable.
Yeah, Chuck, so is that what you're saying? Trying to get the holdfast would be the optimal thing?
Yes, if you could, or at least if you can't get the holdfast, at least get as much of the stalk down to it as possible.
Right, okay, that's what I thought you said. Yeah, so as much as the stock as possible. So I think we might need one that's a little smaller. Yeah. See a Metallogorgia colony standing up there on the right side, going down the slope. So, is this like a faulted area, or am I just reading something into this that isn't really there?
I think this is probably not a faulted area. I didn't really see any signs of faults cutting through the edifice. This is probably a little volcanic ridge, which now is coated with this manganese crust. Would be my guess.
Got it. Thanks. Let's get a snap zoom on the coral to the left, lower left of the lasers, the whitish one. I think this is going to be a Chrysogorgia, the sort of bottle brush morphology, and we haven't seen that genus today, I don't believe. Yes, indeed. And so this one's a little bit more compressed. This reminds me of the species Chrysogorgia chryseus. So that's a possibility. And it's got a squat lobster tucked away down in there amongst all the polyps. And I'm seeing these polyps also look swollen. You can see there's a more peachy color, quite round. So they're kind of packed with eggs as well. And Chrysogorgia is one of those colonies, one of those taxa, one of those genera where it's very, very common to see these squat lobsters in the branches. Tim, if you're there and you want to say anything about squat lobster relationships with corals, please feel free.
Yeah, Scott, this is Tim. This is a classic Chrysogorgia morphology, and it's a classic position for the chirostylid crab as well. I was just thinking that we haven't seen any chirostylids today. This is our first one, and it would be appropriate to see it in a Chrysogorgia. It's always difficult to try to image these things and see what get some characteristics from them. I'd love to see the front end of them, or at least the chilipeds, the claws, be really informative. But it's good to know that they're here.
Thank you, Tim. Yeah, they're rarely cooperative. They like to tuck down in there, and it's no surprise. They're presumably using the coral as a place to protect themselves from potential predators.
Will they leap out of these if you disturb them, or do they hang on?
They tend to hang on.
Uh-huh.
Pilot, if you have the time, you can look, but if I don't want to reposition or hold the ship up just to move to get a position for the crab. Okay. So they show a lot of fidelity to the colony. And so they would rather be here than elsewhere. So they rarely, the chirostylids will rarely leave. So when you do make the collection, you almost always get the crab as well.
Interesting.
And, there's a great example we had of making a collection that had a chirostylid on it. And we were interested in the crab. That's why we were collecting this particular coral. It was actually a whip bamboo coral.
Okay.
And the chirostylid came off during the collection. And so we moved the colony back, or the pilot did, behind the crab. The crab scuttled back up and grabbed onto it. So they got it. Yep.
That's cool.
Their back legs are strongly hooked and hook around the branches, and they just hang on there. Better adapted to be in branches than on this smooth rock.
Right. It's interesting. And do we think they stay on the same colony all their lives?
That's my hypothesis, and this is one of the things that Tim's been studying. First, what's the association with species? Do you have one chirostylid species per coral species, or does it not matter all that much? And then the secondary question. Yeah, so you're thinking of a different Chrysogorgia species. These chryseus are really thick, and they're much more compressed, so it's hard to see through all those polyps. Not the first to get fooled by a Chrysogorgia. Probably not the last. Tim, feel free also to make a comment on this. I know that in some of the black corals, we often see 2 chirostylids, and one's a male, one's a female, and the thought is that, you know,
right.
that's home.
They've taken up residence there as a couple.
Exactly. Looks like a dead sponge there.
Yeah.
Lots of barnacles again.
Yeah, no end to the barnacles.
So, you know, if any of...
Quick question, Mr. Chuck Messing. I just want to know how deep you are.
Oh, yeah. Sorry, we haven't said that in a little bit.
We're 1653.
2.7 degrees Celsius, Chuck. And plenty of oxygen, 3.2 MIGs per mil. Oh, there's a sea star right here above the lasers. Yeah, we can have a quick zoom on that. We may have seen it earlier tucked under a rock, and Chris later gave us an identification. If this is the one that had the prominent spines coming out of the arms. Yeah, exactly. Yep. Yep, thanks. So I think the second, at least the second one of those we've seen today. So where was going just before that? Oh, I was going to say if you've gone to the seashore, a rocky seashore, you're probably familiar. Let's get a quick zoom on the colony right at the lower center. It's whitish and there's something red in its branches. I think this is the second primnoid. Yep, right on the lasers. Yeah, so we saw this earlier. So this is in the family Primnoidae. And it also had one of these ophiuroids, and Tim Shank had called in a little bit later to say that this similar morphology was what we saw on the plexaurid, and we thought that was kind of interesting that it would cross across those very different families. Thank you, pilot. No, that's good. We had a closer look at this earlier. Yeah, so if you've gone to the rocky seashore, you can see lots of barnacles on rocks and mussels and limpets, and the thing is they're all really low and squat so that they can survive a lot of wave action. So it's interesting to see all these barnacles and fewer tall things.
Right.
You know, does that say something about the tides crashing across here, the internal waves, you know? I don't know.
So I haven't noticed any of these barnacles open. It seems like they must have been closed. Is there some rhyme or reason to when they open and close? I mean, you would.
Not to my knowledge.
They seem all to be closed at the moment. But
so the one thing, of course, is when they're open and kicking their feet out, that requires energy. They're expending energy, moving the feet back and forth. So I suppose if they can detect that there's not much flow right now, maybe not much food, might as well hunker down.
Settle
perhaps.
down. Okay.
Looks like some dead corals around here and some crinoids.
Yeah.
So maybe once upon a time there was a stronger current here and not so much anymore. There's another purple holothurian tucked down there in the rock. We've seen 3 or 4 of those individuals today. Holothurian is a sea cucumber. Very few fish again. I think Astrid said the other day that you'll find more fish where there are more pools of sediment because the fish are preying on things in the sediment. Just passing over another yellow sea fan. We imaged one of those about half an hour ago. The family Plexauridae, perhaps a Paramericiid. There's another one. Several more of these crinoids. They certainly get to a robust size. Yep. It's always interesting, and that happens a lot with the corals too. You see these big corals, and you always wonder, where are the small ones? And it's suggestive of recruitment happening a very long time ago. And so that begs the question, well, why isn't there continuous recruitment? Why don't we see all the different ages, from the very young ones that have just settled that are 1 or 2 polyps to intermediate, growing larger, and so on? This might be something different.
About the Chuck Metzing, about the recruitment, I saw the same thing in the Bahamas. We get meadows of crinoids, and we never see little ones. Very strange.
I'm sorry, Chuck, I was communicating with the pilot. Could you repeat that?
Yeah, I just said that along the same lines, because we're only seeing tall crinoids here too, in the Bahamas in 400 meters or so, there are meadows of a different kind of sea lily, but we never saw, in 8 years, we never saw little ones.
Yeah, isn't that odd?
I would have picked up.
We're getting a close-up here on a precious pink coral in the family Coralliidae. We collected a member of this family on our last dive. I don't know that it was the same species, but it was the same family. And in the Pacific, you may have heard of Corallium. I believe it's, I'll have to think about it. I can't remember the species name, but there's a Corallium in the Mediterranean that has been harvested for some 2,000 years for use in making jewelry. So these are closely related, and you can see that it also has one of these ophiuroid brittle star associates in its branches. These things are really quite brittle in a sense. They appear to have a hard skeleton, but it's a cemented skeleton. So instead of that axial skeleton like the bamboo coral or the sea fans, it's unidentifiable, so that when you break it open, you know, you don't see any central rod. Instead, it's all cemented together. No, this is good. Thank you.
Just up to the right, there's a small sea lily, but it's only got, looks like 5 arms, so I'm not sure if it's a juvenile of the big one or a different species.
Yeah, Chuck, I was going to ask to pull back. I noticed that there's one of those 5-armed purple or brown ones. So there's going to be a crinoid. I think if you turn a little bit to your right. Oh, there's one on the left, but I thought maybe there's a closer one on the right side. Maybe if you just turn a little. Yeah, there's an Anthomassus there. I thought it was just down below, not up at the Anthomassus level, but below. Huh. Oh, I thought it was there. Maybe not. I might have been making it up. But we can look at the one on the left. Chuck, we're going to zoom in on this crinoid on the left if you want to describe it a little bit.
Will do.
Oh, I see what Chuck was talking about. Right up here is a really small one. Yeah, yeah. It's another one of the urchins in the top right corner. He's getting ready to go down a slope.
Yeah, maybe we'll catch it here.
What's going to happen is we're going to zoom in and look at this one, and we zoom out and he's going to be down.
He'll be down at the bottom,
Yep.
right? In mystery.
Yep. No, there's... There's another smaller urchin just off to the right side there. Okay, I see 5 arms. And Chuck had sent an email before the cruise started. They were looking for a 5-armed sea star, but that had naked tips to the arms, and this does not have those.
Now this one is looks like Echinoderms. It's not the. I don't think it's the new. Well, we don't know about the arms of the new species, but It's difficult to tell whether this is that weird new thing, but I don't think so. I think this is a species of Pentametra crinus. There are several in the Pacific.
Thank you, Chuck. We're going to move on, pilot.
Yeah, still very little sediment up here. Lots of barnacles.
So when we were on the last dive and we were going through the saddle towards the base, we saw those heavy rippling,
right?
Which I think was indicative of a pretty strong current. And we're the same kind of position now. We're just the edge of the saddle and it's starting to go up the slope again. So I wonder if it's really strongly scoured here. I'm not sure what this is, pilot. So if you're as curious as I am. Is it a hedgehog? Oh, is this a crown of thorns type sea star? Or it's an urchin? No, it's a really big urchin on its side. Yeah, I think this is an irregular urchin. Yeah, I think so. Yeah, let's start from the side. How good are you on your urchins, Chuck? This one looks like it has a brittle star on it. Underneath. Maybe two of them. It's just like a, it's just a, yeah, maybe it's a sea biscuit. Nah, the spines are too long for a sea biscuit, I think. There's sea biscuits and sea potatoes. Those are other kinds of urchins that are normally kind of burrowing. I swear to God.
Yeah, I've never heard sea potato, but I think of sea biscuits as being something that are in the sediment, not,
they're
not on a rock.
Flatter.
Yeah.
Yeah. Yeah, so this is interesting.
So how is he able to hold on there?
Well, presumably the tube feet are hanging on at the other end. So you can see there are two brittle stars tucked on, taking a ride along with this thing. I guess this tells you the power of the tube foot.
Yes,
how well it can hold on to the rock.
If you've got a few hundred tube feet, you can do anything.
Diva Amon suggested Echinotheridae. We saw earlier another urchin she suggested was in this same family, but it was in a more typical position where we see it from above instead of perched on the side like this. This is a pretty good size. Did anyone notice as we were zooming in what the size was? There's our lasers. So it's almost 10 centimeters from top to bottom. That's a good size urchin. Some of these urchins have toxins associated with their spines, and I know from experience that when you pick them up, that is not a pleasant sensation. There's a little scale worm on the substrate behind it. Thank you, pilot. That's a nice observation. So urchins and some asteroids have structures that are called pedicularia. And they're little stalks with ossicles, the hard parts that have some muscle and articulated, and they're kind of like little tiny pincers, and they can hang on to little bits and pieces of material. And I wonder if the ophiuroids are taking advantage of maybe some of the organic particles that are being held by the urchin, and so feeding off of that, or some bacteria that's associated with it. It's really easy to come up with these ideas, questions and hypotheses. It's really hard to go and test them.
So, one of the tools we have more and more at some active spreading centers, we have these underwater cameras now on some cabled arrays. Has there been much sort of long-term deep sea photography that might capture some of these behavioral things here?
Sure, and there are different groups who are deploying long-term camera deployments at some of these long-term ecological research stations in the deep. And absolutely, you just got to have them pointed the right way.
That's the trick.
Right, and maybe with something that's sessile, then you know that it's going to be there. Oh, really? Okay, that's interesting. So if I don't know if you heard that on shore, but the pilot thinks he saw a school of fish, and we're going to try to find that.
Was that in the Sirius cam?
He saw it in a manipulator camera.
Okay. Interesting. Uh-huh.
Okay.
Huh.
Could be squid as well.
Yeah, we don't typically see schooling fish at these depths that I can think of.
So Ken Sula, I'll just point out that that is a Brisingid asteroid, and we've seen one or two today, but I don't know that we've really made a good note of them, but it's something that's very common that we see. So that's a sea star that sort of feeds on the suspension, the stuff floating in the water. So Ken Sulak, who's a fisheries biologist at the USGS in Florida, and he's been providing some information on some of the fish identification that we've seen, has just made a really interesting comment on this question about the recruitment and why we're seeing apparently a whole bunch of these individual corals and crinoids that appear to be the same old age, and we're not seeing many recruits. And he says that it's rather common in nature for organisms that live to be hundreds to thousands of years old, but which have high annual fecundity to have this kind of pattern. So live oaks, redwoods, bald cypress tend to occur in old mature stands with no evidence of successful recruitment for long spans of time. He's been watching this in live oaks for 23 years in Florida, and in that time span, live oaks that produce millions of acorns annually per tree have had only 1 year in which the seedling actually sprouted. It survived about a year and then it died. So the conditions for a rare, successful recruitment event must occur only at wide intervals of year, decades, and centuries. So perhaps it's not surprising that we're seeing the same thing in these deep-sea gorgonians and black corals. Thanks, Ken. That was really interesting. So we are definitely headed up this ridge crest. At least that's what the map is telling us.
Maps don't lie.
Maps don't lie.
Not our maps, at least.
Not our maps, right. The view you're actually looking at on the camera is looking kind of crossways across this ridge crest towards almost just a little bit north of east, almost due east. But the ridge crest that we're following is moving up to the northeast. So once we turn back to port here, you'll see the slope going back up. Del, you don't think we can just reach in that hole and grab a rock?
Oh, well, I don't know what we'll find in there, but
one of the real treats of being involved in an expedition like this that has telepresence, has a number of other scientists that are communicating either over the phone or in the chat room, is you get to learn a lot from each other while you're observing together and exploring together. And so many different experts that can offer insights into different aspects of the biology or the geology or whatever it is, their specialty. And when it bleeds over and you haven't thought of something and you see it from that perspective, it can be really helpful to your own research.
looks like another dead sponge there on the left. And
Another dead Walteria.
Is that one off in the distance as well, Shirley, up in the upper left?
Yes, Amanda, I think so.
Yeah, I think that was the same urchin, yes. And then we were just commenting on a couple of the dead sponges here.
Chuck Messing, any chance of zooming in on that curled up yellow feather star just to the right of the dead sponge?
I think we just went past it. But
Stand by, Chuck. Oh, there it is, the base of the screen, center. You'll get a good view in just a moment, Chuck.
That is Chuck again. Yeah, that looks like a member of the family Charactomystidae, C-H-A-R-I-C. They're typically yellow like that with 10 to 40 arms and widespread at these depths from a few hundred meters or 150, 200 meters down to over 1,500 meters. And the number of species is right now anybody's guess because they've separated out on the smallest characters.
Great, thank you, Chuck. We're also seeing, as we sit here, Pilot, we can move when you're ready, a Polychaete worm swimming by, and there was yet another snail. And there's a dead sponge just covered. Is that the one you were talking about before with these brittle stars?
Yeah.
Why?
Make for very nice brittle star habitat.
Sure. No room at the inn.
Chuck, I was wondering if you know why they curl like that. So it seems like a different morphology or different kind of behavior than we've seen.
Curl up. That's the same kind of, it's a different response to the same conditions as the sea lily wilting like an umbrella. A lot of feather stars, when they are subject to weak currents, they'll just curl in the arms like that.
Okay, cool, thank you.
I'm starting to see patterns in all these barnacles here. It's like constellations or something.
Sending you a sign, is it, Del? Spelling something out?
I won't say what it says. So.
Yeah. Right. I believe it's spelling out, this is not the rock you're looking for. Thank you, thank you.
Definitely.
See how I did that? So again, interesting, fascinating. So we're up on this crest here, and I'm not seeing any large bamboo corals. No. There's a few of the crinoids, and we just heard from Chuck Messing a couple of times saying that when they're in this orientation where they're more upright like an umbrella, it's a good indication that the currents are really weak because they're trying to now capture particles that are sinking straight down in the water column. Instead of when you see them kind of nodding their head forward, that's an indication that the current is strong enough that the water is moving horizontally. So I guess the weak currents are why the corals aren't successful right here. Is this a sponge just to the right of the laser? Oh, there's a fish too. There's a fish right across that dark cavity, I think.
Oh, yeah.
Really?
No fish.
Oh.
No
Yeah.
fish. I meant to say a Galatheid crab in the cavity. That's what I meant. Could we rewind the tape?
So excited.
Sorry, Astrid. All right, we can go back to that.
The rock fooled you, see? Or,
Yeah, I know, the rock. Cursed rocks. Huh. Is this what we were going into? Oh, there it is. So maybe it's not a sponge. Maybe it's a coral. I can just identify all kinds of things. It's okay if we don't zoom on them. This is interesting. I wonder, could this be an Echinella? That would be outstanding. It's probably a Chrysogorgia, but I just want to make sure. Looks like there's another Galatheid tucked in there. So we were saying earlier, we were looking at one of these Chrysogorgia, Chryseis, or Stellata. It's one of the two, but it's almost like they have two fans compressed together. And I think the crab is tucked inside in between them. Just want to get a good look at the skeleton and be absolutely sure. That's what I'm looking at. Yeah, if that doesn't take a lot of time, we can try that. Yeah, Les Watling believes this is a biflabellate Chrysogorgia. So it's, I mean, two parallel fans really close together. Les, I would just wanted to verify that it wasn't an Echinella. And if it were an Echinella, that would have a different structure, more like a Christmas tree, where there'd be more of a couple of central branches that the branches would be coming off of instead of these fans. We call that world branching. This is a good view. There we go. Yep. So Chrysogorgia, thank you. And I think there's actually two crabs in there. You can see two reddish spots with slightly golden dots. Those are the eyes. So there's two crabs tucked up. So that was a good view. Two chirostylids. Thank you, pilot. For whatever reason, these biflabellate ones have more robust polyps. I was referring to Chrysogorgia being so delicate. These ones seem a little bit more strong and robust. There's an Anthomastus, the red mushroom coral. Astrid is also noting that these barnacles. Yeah, go ahead.
Yeah, this is Les in Hawaii. I just wanted to point out that one of the odd things that we catch a glimpse of every once in a while is that it looks like there are a lot of the little stalked barnacles that look like Glyptolasma or something like that actually on the surface of the rock. And I don't recall ever having seen them on the rock, or at least not this abundantly on the rock. Usually they're on the dead calcareous skeletons of the bamboo corals or something like that.
All right, thanks, Les. I'll keep an eye out for that. Right now, I'm just seeing the acorn-type barnacles. I'm not seeing any of the lepatomorphs, but I will definitely have a look. Feel free to chime in if you see one, or some, and we'll get a better look at it.
Yeah, this is Tim. I agree with Les. We've seen them sort of sporadically throughout the dive, including, I think, the very first coral that we imaged. I saw a ribbon of those barnacles on the seafloor, and yeah, I agree with you, Les.
Yeah, Tim, now that you say that, I recall on the ledge where we were seeing the Stoloniferous and all of the young Anthomastus, there were also some there. There's a rock sea pen, the brown one, that's about, yeah, just above the lasers now. Just get a quick zoom. We've seen this a lot, but just to confirm that's what it is. Del, so this is great. So here's the sea pen that has been adapted to live on the rock, and you can very clearly see the peduncles. So that's what would have been buried at the base of those whip-like Halipterus we saw earlier in the rippled sands.
Great.
And it's why it was so surprising to everyone to see the sea pen on a rock. It's just not the right place. This is probably the genus Anthoptylum. You see it's got that kind of question mark appearance. I don't know why it is, but...
Crinoid off on the site? Because that looks like a juvenile of the big one.
I'm sorry.
Unless you've gone past it already.
Yeah, sorry, Chuck. Didn't miss it, and you're pretty delayed, so I'm not sure where you meant. But when we zoom out, I'll have another look. And then there's
shrimp.
Yeah.
Just past that purple urchin.
Okay.
Off to the right of what you're looking at now.
All right, we'll check it out. That was a mysid shrimp, and then you can see the squat lobster in the back.
Are those some of the white barnacles there in the background on that?
I don't think so. All that stuff right here?
No, down light there.
Yeah, no, those are dead acorn barnacles.
Oh, those are acorns still. Okay.
Scalpelids, I believe. As we zoom out, we just want to look to the right to see if there's a small stalked yellow crinoid. And he said that it was next to a purple urchin, I think. Was it a purple urchin or a purple cucumber? I'm not sure how delayed Chuck is by what we're listening to. I think he's on like the 30-second delay, so it might be well past it. Yeah. Okay. Let's keep going, and maybe he's referring to this one right here. We can zoom in on this crinoid right in the foreground. Yeah, I think this is a smaller version of what we were seeing. Unfortunately, it doesn't have any of those associates on it, which is interesting in and of itself. It suggests that it has to be around a long time for those others to find them, perhaps.
Yeah.
You can zoom on the crown. Thank you. Just trying to verify if there's anything else living on this individual, and I don't see anything else. Really see those tube feet. They're very long and fine.
Yeah, this is I'm a little out of focus here, so I can't really tell, but I would guess that this is probably the forking in the arms out away from the base. This looks like a juvenile of the big one.
So Chuck, I'm not seeing any associates in the arms, which is one of the things I think that was exciting about the previous collection. So I guess the question is, how important a collection is this if it was just alone? Recognizing that we only have 2 collections remaining. Are you still there, Chuck?
Oh yeah, I wasn't sure of the delay. Sorry. Yeah, it's that's true. If you're limited. I saw one little whitish thing down at around 6 o'clock out on one of the outer arms.
Yeah, I see what you mean. Yep.
I don't know if it's the same thing, but this does look like a juvenile of that big one, because you can see the forks in the arms are way far out, which is unusual. But I leave it up to you guys.
We need the expert advice to know what to collect. I just want to make sure that we're using our collection wisely. If this is something that you've
Right.
been looking for.
Well, it is certainly. I mean, we only have the one specimen. That would be the holotype, and this would give us a different size image. But you see, as it comes around there's one white little item down at the bottom, which may or may not be one of those quote-unquote parasites.
Myxosomas, yeah. So, Pilot, I guess the question is, how easy, given the position that we're in, to get this from the base? And that was the critical thing. We'd need to get most of the stock. Yeah. If let's see if it plucks if you do the grab, and if it doesn't, then we'll do the trim.
Thank you very much.
Chuck, can you spell the genus name for me again, please?
Yeah, as far as we know, it's P as in Peter, H as in Harry, R as in Robert, Y as in yellow, N as in Nancy, O, C-R-I-N as in Nancy, I, D as in David, A-E. I'll go to family.
Okay, that's good.
There are two genera in the family. It's probably Porphyrocrinus, which is like porphyria, P-O-R-C-H-Y-R-O, Crinus.
I think I will, for the sake of the field ID, I'll stick with the family name. But I think I missed something, P-H-R-Y, and then I missed something before I got to the O and the E.
Okay. P-H-R-Y-N as in Nancy, O, C-R-I-N-I-D-A.
Great. Thank you. Got it. Go ahead, Watchley. Go ahead, Pilot. Excuse me.
Of course, it may be a new genus, too.
Chuck, I'm also going to be sending you an email asking you about any particular details about preservation of this specimen.
Roger that.
So the magic of telepresence, we are here in the Central Pacific, just about 90 miles south of the equator, while Chuck Messing is on a ship in the Caribbean, in the Bahamas, doing his own sampling and is able to request a sample from the other side of the world. That's pretty impressive.
That's pretty amazing, isn't it?
Yep, it closed up, so you can see that it is capable of sensation, can detect its environment, and closed its arms upon contact with the manipulator.
This is Chuck again. That's an interesting point about collapsing its arms because on the inner side of the arms is where the muscles are. And the only time it needs to use those muscles is to collapse like that. The elastic ligaments, which actually have some contractile ability, are what uses to spread its arms out against the current and hold them there without getting exhausted.
One of the really cool things about these echinoderms is that specialized collagenous material that holds these articulating ossicles together. And as Chuck was just saying, they can be basically solidified or liquefied. And so you can put the arms in a particular position and then fix them there basically by solidifying that equivalent of a ligament. And so you don't have to have any energy. You don't have to be using any energy to be holding your arms up into the water. Seems like a good strategy for something that lives in the food-poor deep sea. Why do these things always have to grow in such a difficult position to collect?
I'll take what we can get.
That's right. Yeah, it's you know, as typical, it's not on a really flat surface, and so it's really hard to get the manipulator claw down in there. And so we're able to get about, I would say, 1/2 to 2/3 the length of the stalk.
Outstanding, gentlemen. Thank you very, very much.
We'll look forward to a report, Chuck.
All right.
Nice job, pilot. The sample is in the box.
So yeah, that's our
So
fourth,
starboard inverted biobox.
yeah,
Okay.
fourth sample of the day. We got a piece of coral. Then we got some sediments with the scoop. And we got a rock with a little urchin passenger on it. We just picked up a crinoid, so filling it up.
So a reminder, we are at 1,636 meters, and I think we just noted that we are diving just south of the equator, 1 degree 30 minutes south of the equator, and we are in the boundaries, within the boundaries of the Pacific Remote Islands Marine National Monument, the Jarvis Island unit, and we were diving on a seamount that was only just mapped last night by our mapping team. At 5 a.m. this morning, we were looking at the fruits of the labors of the team that worked overnight to collect the multi-beam sonar data and the backscatter data to make that map, and we planned a dive site, and we were provided with a name. We're calling this Kahalewai seamount in honor of some of the Hawaiian colonists who were living on these islands back in the 1930s. And we heard an interesting historical lesson on how these colonists were brought to the islands and the reasons for that. And there's a really nice essay on the website if you would like to review that and read it. But in particular for this seamount, it's being named after Carl Kahalewai, who passed away while living on Jarvis Island. That was back in 1938. This is interesting. What's going on with this crinoid? I imagine this is a dead crinoid. Maybe all its juices have been sucked out by... I better not see any interesting stuff on that crinoid on the right, so I don't even want to look at it. Let's just look at the brown one. I'm just kidding, of course, when I say that.
We get it in the same box.
Oh, this is a different species. It's in bad shape. Now, Chuck, I don't know if you're going to see this one. It's brown in color. It does look like there may be some branching of the arms towards the end, but this one now is covered in those yellow things. And this one has the heavy rings, the ring-like ossicles on the upper part of the stalk underneath the calyx. It does have at least one.
Wow.
Yeah.
Yeah,
Exactly.
he doesn't look so happy, does he?
No, it doesn't. You think this is the same species?
Well, if we get, I mean, the arm branching pattern is the same. It really doesn't look very happy, does it?
No, it doesn't look happy at all, and it's got a number of these things on it. I wonder, you know what, I'm starting to think these are aplacophorans. So as I look down here, it looks like I see what looks like a folded foot over one of the pinnules. I mean, they're not like any aplacophoran I've ever seen. Have you ever seen any on a crinoid?
I'm sorry, I didn't quite get that comment about.
I was asking if you've ever seen an aplacophoran mollusk on a crinoid.
Oh, no, I don't. That's one group that I'm not familiar with, but sure, why not?
Yeah, exactly, why not?
I don't know if it's ever been found on crinoids. But on this close-up that I'm looking at now, there's towards the bottom there's a reddish swelling, which looks like a gall of some sort. But why not?
Yeah.
You know, almost everything else makes a home on a crinoid.
Yeah, they kind of look like selenogasters to me. Interesting.
That would be interesting.
It sure would. So selenogasters are kind of a placophorine mollusk, again, worm-like mollusk, and they have a kind of a groove where the foot of a snail would be, and that groove could kind of wrap around these pinnules that you're seeing, these little side branches from the arm. Very interesting. Whatever it is, maybe we'll find one on another dive. Yep. Go ahead, Chuck.
There are a couple of darker ones, and the one right in the middle looks like it's plated, and that's typical for a gall caused by a mysostome.
Okay. Well, we certainly know that mysostomes would be the more expected thing. Yeah. So mysostomes is this strange little group of Polychaete worms. In fact, it took a long time before anyone realized they were actually Polychaete worms. It was unclear what group they belonged to of invertebrates. And so it may be that they're inducing the crinoid to form a gall, just like the way some plants and trees form galls around parasites that are associated with them. So maybe that's what we're looking at. Thank you, Pilot. Very interesting.
And also, a lot of the arms are broken, and there's regeneration, white regeneration at the tips of some of them.
There's all kinds of brittle stars that are living on the lower stalk. I mean, I guess that's
seem to be weighting it down there.
Yep. I don't see a bright future for that crinoid. Oh, great. We have a notice here in the chat room from Kevin Kokot who says there's no described selenogasters feed on anything but cnidarians or occasionally annelids. So he believes the brownish mass is endomysostoma, and the others are different mysostomids. So these would be these unusual polychaetes that are known to be parasites of crinoids. So that makes a lot of sense. Thank you, Kevin. I really appreciate that input. And it looks like we're coming up to another interesting bamboo coral that may have a fork in it. So again, these whips, it's in with a cluster of barnacles. And we see a couple of crinoids on a cleared bit of skeleton here. So my usual request, if it's possible, to get a look at that supposed branch point. And again, so what I'm looking for here is evidence that this is truly a branch, or could a larva have settled on the existing colony and started a new colony? Or was there an injury and the regrowth precipitated off in a new direction, that kind of thing? And if that's the case, you would see that there's going to be some kind of a swelling or something at the branch instead of just a smooth transition.
Not just a simple branching, but something more complicated.
Yeah.
Well, let's see what we can find.
Otherwise, this would be a bamboo coral in the genus Lepidisis. You can see a bunch of barnacles also at the top of this, where the crinoids are hanging on.
Quite a few up there.
Yep.
Yeah. We had seen some at the base earlier of some of these, but not that far up.
Yeah. That looks. Oh no! You see how thick it is right down below? Look at that.
Yep.
So it's really thick and
very thin on both sides going up. Looks like
So I wonder, there may have been something that occurred at the top there, and then it just bifurcated around whatever it did. Thank you. That's good. Essentially, it's going to take several different collections of different kinds of weird branches like that to try to unravel that mystery using genetics. Yep, here we go again with a crinoid with apparently lots of mysosome polychaetes on it. We don't need to have a look at this. We've seen this several times now. Thank you. See how high we can get up on this slope. It's a much smaller crinoid that we're passing over down there. It's not even anything on this edge of the slope or the ledge.
That was a big piece of rock sort of dislodging here on the slope.
Seems pretty clear, doesn't it? Do you want to see if there's anything loose in there? Doesn't look like it.
No, it doesn't look like it. I don't know if we can get down there or not. Probably not.
A little bamboo coral starting to grow right there. It has 3 points. The laser's just passed over it. And I see another fuzzy little Chrysogorgia colony on the front of that big rock that seems to have separated from the right side. And what is this? Arms of something just around the corner. Maybe tips of another, maybe a bubblegum coral. I believe I've been wrong on calling a bubblegum coral every time we've seen one. Not actually confident we've seen one yet. Oh, look at that. There's a yellow bamboo coral tucked in that hole.
Yeah.
That's odd.
You wouldn't think you'd get a lot of flow through there, would you?
No, but you know, you got to figure it's the larva that's doing the settlement, right? And so what does it know? So some stupid little kid finds a place to settle, and later on as an adult it finds that, oh, I'm in a cave. Yeah, please have a close look at the red.
I hope the kids are listening out there. Okay.
Oh, this is good. This really is a bubblegum coral, but it's got a special treat. That left laser is lighting up something. Yeah, do you know what that is? Anybody?
An egg?
Yes, that is
egg.
an octopus egg that Caitlin has just pointed out.
We saw one of those earlier on a dive, I think.
Did we?
Yeah, I think we saw it once.
It's
Yeah.
ridiculous. It's literally like the same things are being seen on all of these cruises. We saw, like, this had never been seen before, right? Before the last expedition. And now here we are again. Pelagothuria hadn't really been seen before the last expedition, and then we've seen loads of them. What is happening?
Well, what's happening is maybe we're going to get more people interested in the Central Pacific and get more expeditions out here, Diva. There's also I think a hermit crab down below. But yeah, so this is really interesting. You may have seen photos, or you can search for photos of what they call the Dumbo octopus. And it's a small octopus that has fin-like structures that make it look like ears of Dumbo the elephant. And we have seen, certainly in the Atlantic, and now as Diva points out a couple of times here in the Pacific, that they lay their eggs on corals. And so they have this sort of outer organic sheath. And I would imagine this one's pretty far along. And at some point this will hatch. And
so we just sit here and wait till we have a baby octopus hatching? That's gold right
so
there.
I'm glad you said that, because there's an interesting paper about an octopus that they found in Mbari who was brooding her eggs. And that's one interesting thing. Many octopus brood their eggs or lay them into these mats and hang around for a while, and then they die. Well, this female stayed with those eggs for 4 years before they hatched. Not feeding. She was not feeding.
I don't think we have that much bottom time.
We don't have that much bottom time. But otherwise, this is a bubblegum coral, and you can see the swellings there. The polyps are retracted inside those swellings, and you can see why it's called bubblegum, because it looks like wads of red bubblegum. Thank you. That's new for the cruise. Great observation. And there's another little hermit crab just above it. And you see that brown wormy-like thing that's on the rock? That is an aplicophoran. So that is a worm-like mollusk that is a predator. And there's some barnacles, another snail.
Yeah.
I don't know if those are snails or if they're hermit crabs, but that's good. Thank you. There's some of that spiderwebby stuff that Astrid was talking about. Boy, this is a really interesting dive site.
And Shirley was just pointing out, it's just in time for Octopus Friday.
Oh, my goodness.
Oh.
We had Octopus Friday last week. We discovered an octopus for you.
So
maybe it'll hatch tomorrow on Friday.
And it's already Friday in New Zealand.
This, I was going to say maybe a Echinoderm, but I believe this is a larvation house. Yes, this is a larvation, so I don't actually see a larvation in it. So a larvation is a small pelagic tunicate. It looks like a little tadpole, and it secretes and builds these mucus houses around it. And what you're looking at is the mucus house, and you can kind of make out that there's some filters in there. And basically the animal lives inside there. It beats its tail to generate a current, which draws in water and all the particulates associated with it, and it feeds on those. And then once the filters get clogged, it ejects that house, and it builds itself a new one. It can do that several times a day.
Several times a day?
Several times a day. And one of the things that's so interesting is that The house then becomes clogged with all the organics. That makes a great food source for lots of other things. So it's a concentrating mechanism of organics in the water column. Thank you. Good, good eye. Amanda points out that there was an article about larvaceans in the New York Times yesterday, so they've made the big time. Got their press agent out there.
A day late here.
There's another, I think, Chrysogorgia, those biflabellate Chrysogorgia colonies down there. Barnacles on the top of the rim. Tim Shank from Woods Hole is noting in our chat room that it's interesting that the octopus egg that we saw was in basically the same state as the one that was observed on the last ROV leg. That is, the outer casing, that sort of green-brown soft stuff that you saw, was peeled back to reveal the inner purple membrane. What's this floating? Just looks like some mucus, and I'm not sure what else. Doesn't look alive. Yeah, no, just a mucus string that could come from any of a number of things, including squid. If there are squid in the area, and earlier our pilot noted that he saw a number of things swimming out in the water column that he thought could be fish or something else. If they're squid, when they jet away, sometimes they'll release some ink at the same time as mucus, and it forms these kind of stringy morphologies in the water that are called pseudomorphs, and they're good as kind of decoys for as the squid swims away. So it's possible that that's some of that stringy stuff that we're seeing. Tim Shank is noting that the octopus egg that we saw on the previous ROV leg also was on a bubblegum coral. So that's interesting. Looks like a bamboo coral tucked way down there in the cavity on the lower left. But otherwise, there's not too much on the side of that wall, is there? There's something white inside this cavity on the right side. I don't know why I'm kind of surprised at the number of little holes and caves and cavities and crevices there are here.
Well, we haven't seen all that much in them, though.
No.
Yeah, we haven't seen a lot of crabs or anything like that in there.
Oh. Yeah, I don't know what that is. Something started growing and then realized it was in the wrong place. Thanks, Sean. So, I mean, what about the underlying flow? Is this, I guess these look like pillows, aren't they? Is this pillow lava?
I mean, they really do look like lava flows. Now they're all crusted over. It's exactly what we would expect. That as the lava comes down this slope, it's sort of tumbling over itself. We might make these little cavities.
So that hole that I'm seeing just below the lasers now, could that be the inside of an old pillow, or is that something else?
It could be the inside of a pillow. It could be a tube. We can still get tubes in channelization, even underwater. So yeah, it's not. This is exactly what we would expect for a volcanic terrain. But what we're seeing on the surface is this manganese crust, which can take a similar appearance in some places.
Right.
Here's a little urchin.
Yeah. So we've now seen probably a dozen of these today. I'm surprised. Plesiodiadema, I believe, is what Diva suggested as an identification earlier. Thank you, pilot. You'd think that would have a really hard time moving around in an area like that.
So I want to see it go down slope. I think that'd be interesting. So
it's like you get in that hole. I don't know if you're going to get out of that hole. There may be something, I'm a little leery now because I'm pointing at shadows, but purple on the upper right quadrant on the flat surface. Yeah. You're almost, still just a little bit right of the lasers. Thanks. Oh, I think it's one of these purple crinoids, the 5-armed crinoids, and it was tucked down and I just saw 1 or 2 arms sticking above the bottom. Yep, that's exactly what it is. Thank you. That's good. So the collection we made for the crinoid that we got, I think is going to turn out to be one of the representative species of the day, which is really
That's
good.
great.
That's another reason we make these collections. There's certainly these crinoids are very common. See another one tucked down there on the lower
Yeah,
side.
off the
It's a little bit
side.
more beige in color, but it has the same general appearance. Maybe not. That might be different. I don't know if Chuck is still with us, but Chuck, we're looking at a crinoid here. I don't think its arms are branched, but it certainly has a lot of arms.
I'm here, and I guess because of the delay, I'm just seeing the two big ones, the two big yellowish tan ones on the top of them.
Okay, this one's coming, and this is one of the ones that has the stalk with lots of cirri, long cirri that come off the stalk.
Oh, yes. It's down bottom right, bottom left. That's one of two genera, either Metacrinus or Saracrinus,
Right.
S-A-R-A or M-E-T-A.
Okay,
It might be,
Metacrinus.
so another one, Diplocrinus. I'd have to get a closer look.
Thank you. Oh, we can go. Thank you. He's going to get the close-up in a moment.
Yeah, I think this is Diplocrinus, D-I-P-L-O.
Great. Thank Chuck. Diplocrinus. So that would be another different crinoid from the one that we've been observing up to this time. So some good crinoid diversity. We did see some of the non-stucked crinoids earlier.
Yeah.
So we have about an hour and a half remaining on our bottom time in our dive today, and we're making good progress. And Nav thinks we might be able to make it to the top of the peak, or the local peak, I should say, not the peak of the seamount, but the local high on this ridge.
Looks like a sponge in there, in that little hole there. Yeah.
I see Ken Sulak is asking a question. Maybe we can get a close-up of this urchin that's hanging on the underside of the, looks like a collapsed pillow to me.
Yeah, it totally
or a
does.
tube. But what's the difference between a pillow and a tube?
Well, a tube would be transferring lava a lot further. So a pillow would be just a little bud that breaks out. But yeah, this does look like it could be a tube here. I agree.
Okay. Ken, I'll address
And
your.
could you please
yep.
what depth we're at now?
Say again, Chuck.
1610.
Could you please?
Oh.
Depth?
Yep.
What's the depth?
1610, 1,610 meters.
Thank you very much.
Yeah, so maybe Diva can remind me. I don't recall if this is the same urchin that we saw much earlier in the dive, or if this is now a third species. My sense is it's a third species.
Just checking, Scott. But
Okay.
it does look very, very similar to the Cenopadina we saw earlier. So I have to say, I mean, it may not be the exact same species, but I'd have to compare them side by side to say that definitively. But it definitely looks like the genus Cenopadina.
Great. Thank you very much, Diva. Thank you, pilot. Ready when video is. So Ken Sulek has this interesting question. He asks, how do bubblegum corals feed? Because we had that close-up, and you could see those knobs. It was very, very smooth, and you could just see these little slits where the polyps were. And he said that those apertures seem so small, it would make feeding difficult. And so it's fooling you a little bit because we're basically seeing it in its, resting defensive stage. The polyps actually extend from those slits such that you would see the body of the polyp and all the tentacles extended, and then that colony would take on a very different appearance. It would look very fuzzy. So it's just that the polyps are retracted. They don't take in food through the slits. Instead, the entire polyp comes out through that slit, which also seems a little bit incredible because it did seem like they were tight.
Right.
But, that is the case. Thanks for asking the question, Ken, and following up, Nolan. Are these loose?
Yeah, pilot, can you see if these rocks here to the left are loose? Can we get in there, or is that too tight a spot?
Those are the first rocks to me that look at all loose. I don't know if they're too thin.
Yeah, we can get a bigger one the better, but we've had a hard time seeing anything, so we may take what we can grab.
Yeah, I mean those two definitely look loose. Maybe that.
Yeah, that further one might be a little fatter.
Yeah. Okay. Okay. Thank you.
Will do, thanks.
There's another one of those urchins tucked down there to the left of the rocks. Again, how the heck they get in those positions? And another rock pen. Possible genus Anthoptylum.
Yep, Deb is just agreeing, with your interpretation of the lava tubes there, Scott.
Yeah. So here is an Anthomastus where the polyps tentacles have been retracted, but not the entire polyp. In fact, you can see it at different stages. So some of the polyps have been withdrawn somewhat towards that bulbous face. Others, the body is still out, but the tentacles have been pulled in. And then you can see at the lower right are two where the tentacles are still fully extended. So you asked that question about the bubblegum coral, Ken and Nolan. Just imagine that this was the tip of a bubblegum coral and the polyps are being retracted into the main mass of tissue. That's basically the same sort of thing. This is just on a bigger scale. The polyps are much bigger.
Yeah, if we can grab it.
Is there any rock that you wouldn't be interested in, just as a matter of conversation?
No. As a matter of conversation, no. Given how few and far between the loose ones have been on this dive, I think we'll go for what we can get.
Well, that's tucked down in there.
Yeah.
Interesting view on the Sirius cam right now of the D2 in front of this feature. Looks like a little, I don't know if outcrop is the right word. Is that what you would? Yeah, okay.
Kind of wedged in there, huh? Kind of wedged.
I got it.
Yeah.
It's 5, right?
Yeah.
Do you call them anything other than rocks?
Manganese crusted rock.
Okay.
And then rock, yeah.
Looks like you're gonna have plenty of rock there.
Yeah, that's a good chunk there. It's bigger than bigger than I think we thought. I'm gonna watch the ROV tilt starboard here in a second.
Don't even joke.
Yeah. I understand there's a prize for the biggest rock collected during capstone, so that's really what I'm going for. Oh, it just fits.
Wow, look at that. It's like the box is designed for it.
Well done.
Nice job.
Thanks, guys.
Well, after a collection like that, I realize I don't think the front row has introduced themselves, and so when you feel you're all ready, please take an opportunity to do so. Caitlin. Thanks, Levi. It's like another one of those urchins next to what I believe to be a Brisingid asteroid, but maybe a different one. So if we have an opportunity, if we're not too far behind, a close-up on the sea star just to the right of that urchin that the lasers are on. Thank you. This has fewer arms than the ones we've been seeing earlier, and Diva, I'm not sure if you're comfortable with these. I wonder if this is closer to Frayella than Brisingida, but I don't recall. Yeah, so it is Brisingid, so that's interesting. There's something under it on the left side. So this is a sea star, and this is one of the sea stars that's adapted for feeding in the water column. So when it does feed, it would turn its arms upward. The mouth is still underneath against the rock, but it's basically fishing in the water column and would transfer whatever it captures to the mouth. So what's going on in the spines there? Must be a Polychaete worm. Go ahead now. One hour. Copy that, Nav. 1 hour, 15 minutes of bottom time. The high point is not a priority, but acknowledge that we would have to push to get there if we want to be there. Thank you. Understood. Thank you. So we have 1 hour and 15 minutes left of bottom time in this exciting dive. And so indeed, that's a very cooperative polychaete that just crawled out from between the arms and showed us a little bit about its movement. Thank you. Getting a really good diversity here today. We have some Polychaete fans on shore who are referring to this as very cool and adorable. And I believe that was the family Polynoidae. They're called scale worms because those paddles that I was referring to that extend from either side of the body have extensions on the dorsal side that are flattened, and they create a series of scales over the dorsal side of the worm, such that there's a little space in there. And a lot of these scale worms live associated with other animals. So for example, they might live on the inside of a clam or inside of a tube, and they keep some space there, and then they can draw a current, and there's gills in there as well, so they can continue to make sure that there's aeration and oxygen exchange in tight squeeze spaces.
Yeah, I don't know how long we've been going through this barnacle and crinoid forest here.
Barnacle-infested bottom. It's another one of the large sort of, I'm going to call it a pancake urchin. I'm not sure if that's the right terminology, but we've seen several of these urchins today. Surely Pomponi is acknowledging what we've been saying, that we've transitioned from a lot of Octa corals at the beginning of the dive to an area that's been dominated by barnacles and crinoids and perhaps some of these mobile echinoderms as well.
Yeah, when we started, it looked all like our previous dive, but we certainly got out of that.
Yep,
And,
I thought it was going to be another bamboo coral forest, and it wasn't. And so that's kind of interesting. And, you know, we're much shallower than we were, right, when we did that dive where we saw the forest. So maybe we were just at the sort of upper limit of where they live. I don't know.
Yeah.
It'll be interesting to see if we see any more as we go forward. This is one of the nice things about the program, the Okeanos Explorer program, gives us this opportunity to explore, see what's here, come up with some new questions, and then formulate some hypotheses that we can come back and test. So in this case, if our hypothesis is that, well, maybe the bamboo corals that were making up this forest could only extend up to, let's say, 1,600 meters, and then you get a transition into another environment, you can come back and test a series of depths and different seamounts and see if that's the answer or if there's some other related or unrelated factors that are causing the distributional difference.
Yeah, sometimes we need a little data in order to actually ask those questions, and that's the kind of thing we can get out here.
Yeah, I think it'd be very rare to have a proposal funded that didn't have some advanced data associated with it. We're just getting a transition of the crew, the ROV crew in the front row, so we're just hanging here in the water column for a couple of minutes. Is there anybody on shore who wants to make any observations about the dive or anything else while we're making this crew change?
I think it's interesting. Sorry, Scott. This is the Harbour Branch Oceanographic ACC Institute. ACC. I think it's interesting that we've seen a lot more deposit feeders on this dive than we did on dive 3, which had a lot more sediment.
Yeah, that's a good point.
I don't know why that would be, but yeah.
Maybe the deposit feeders have eaten all the sediment. They forgot to extrude it out the other end.
Forgot to make the deposit. Yeah.
Forgot to make the deposit. Just a withdrawal, please. No what? Exactly.
This is Chuck Messing, and I have seen other places with lots of crinoids, but to find a place like this with a species that's never been seen before, and it's one of the dominant forms, is really kind of amazing.
Yeah, Chuck, you know, we've now had a similar observation made in, I think, 3, maybe 4 different contexts that, for example, the Pelagiothuria, the pelagic holothurian, which many of us had never before seen in our lives, suddenly seems almost, I won't say commonplace, but, you know, we've seen it now several times on some of these dives. And we were just talking about those Dumbo octopus eggs on the Paragorgia and these crinoids that you're referring to. So totally agree. And it says a lot about the importance of getting as many observations as you can. Something that you think is particularly rare, if you're in the right place, maybe it's not rare. So then you get these questions about, well, why? You know, what is it that's different about this place from another place that allows this apparently rare elsewhere species to be common? Looks like there's a whip-like bamboo up ahead. Boy, if this is the crest of the ridge, it sure is narrow. I wasn't expecting it to be this narrow.
Yeah, it sure looks like it drops off on both sides here, doesn't it?
Once again, it looks like there's a little fork in this bamboo. Maybe I just have to stop calling it a whip.
The forked bamboo, yeah.
Yeah, well, I do use that term, forked bamboo, and I thought it was an aberrant thing, but I don't know. This one might be interesting because it looks like that second branch is really small. Huh, look at that. Yeah, that's just newly growing. There's not even a polyp on the tip of it. Just like the one up here, you can see the tips don't have a leading polyp growing. And I don't see any evidence of injury there.
Except that it does change thickness, yeah.
Huh. Yeah, I mean, it's a dramatic change in the thickness.
Could you get that whenever you break the end off, perhaps? Yeah.
Yeah, possibly. You know, so we talk about the holdfast, which is where the colony is connected to whatever substrate that it's growing on, and it's often in the form of a disc. But you could imagine if that there were a settlement on top of a colony, it's going to put down some calcium carbonate. It won't be in the shape of a ring. It's
Right.
going to thicken what is below it. So it's one possibility. This looks very normal to me. This doesn't look like there's anything aberrant going on. It just looks like a branch. Thank you, pilot. We have seen several examples in bamboo corals where there are these nodes that have calcium deposited over them. So we know that it's possible to have that sort of, I'll call it secondary growth.
Right.
You know? But what causes it is another question. So this is a view, that's looking to the northeast, right? I thought that was the direction we were headed. I guess the angle of the ridge is turning a little bit more towards the north, I see.
Yeah, I think that's right. We started going northeast. Now the ridge trend is more straight north,
Right.
I think.
Yep.
We're crabbing a little bit here.
Yeah. We did just do a pilot change. If you guys would like to introduce yourselves, and there's been a request from shore that this time you do it on camera. I don't know how easy that is, but looks like it's pretty easy. So on camera 3, if you would like to see your front row pilots.
Sean Kennison sitting in the nav station.
Annie White sitting in the video shading position.
And Caitlin Bailey is sitting in the video clipping station.
And we are all engineers and videographers with the Global Foundation for Ocean Engineering. Ocean Exploration, excuse me.
Thank you all. Guess we might as well get it all done at once. I'm Scott France from the Department of Biology, University of Louisiana at Lafayette.
And I'm Del Bonesteel from North Carolina State University in Raleigh.
Looks like a small crinoid, perhaps. Boy, it's so smooth. Now we're seeing a lot of little white dots. So I wonder if these are really small sponges or something. They're smaller than the barnacles that we were seeing. It's like there's been a hailstorm that just passed through here.
So our depth right now is about 1,592 meters. And
all right, we're going to take a close look. And so we have a stalked crinoid that does have forked arms. So that may be what we just collected. And then we have a small urchin behind it that has the very straight spines, but not the big curved spine like the Polychaete. And then, yeah, Josh, if we could get in some position where we see a close view of some of these white dots that kind of look like snails on that view, actually. I'd be kind of shocked if there were that many snails on the bottom. Indeed. It kind of looks like there's soft tissue coming out of one side of them. There's a snail. That's a nice thing to get a good view on. Yeah, so there's a live snail. You can see the soft foot coming out of the left side. I cannot make out what all this little white stuff is. I just know there's a lot of it.
Could those be, Chuck Messing, could those be spirorbid type calcareous tubes of polychaete?
Right. You know, I don't. Yeah, I know what you mean, Chuck, and I don't see the structure of the tube that I would expect. They're much more rounded. I mean, they look a lot like little snails. Here's one that looks like it's turned over, and you can see what appears to be an aperture. I don't believe they are snails, but I'm just using that as kind of a descriptor.
Right.
Well, I think it's fair to leave a few mysteries in the deep sea, so let's move on. Certainly a lot of them. Urchin's calling it a day and heading home. Oh, yeah. There was some kind of eel-like fish that was just above in the Sirius view. Yeah, it does look like a gulper eel. It's kind of big and thick at the head end, and then a very, very long tail. Let's see, so what's the heading here? Yeah, so we're going almost due north. We're just a little east of north, 17 degrees, 20 degrees. That's the view that you're seeing out of camera 1. It's so narrow. So when we talk about diving on ridges and ridge crests, and one of the reasons we like to do it is because we think it accelerates currents. I mean, this is exactly what we want. This is what we would think from just looking at a map would be a perfect place to come and find corals. And, you know, they're not there. So we haven't got everything quite figured out about where they are. And obviously, you know, it has to do with the rate of current flow, the amount of particulates.
Yeah, either the particulates are low or this area is being swept clear of sediment. We haven't seen anything up on the top of this ridge,
right.
no sediment whatsoever. So either it's not coming down or there's at least enough current to move it off. Even in the little crevices, we're not seeing it here.
Yeah, and we know there's enough current because there are some corals, like the one that's coming up ahead here, another forked bamboo. And there are, you know, the crinoids and so on. But it's not a forest, so that's interesting. We do think there's one of those paratrooping
heading
brittle
down.
stars.
Yeah.
We do believe overall this ridge is kind of in the lee of the dominant current, so it's possible that the 900-meter peak ahead of us is having some impact on this ridge. But as you say, there's no accumulation of sediments here, and we certainly saw some down lower, down deeper, I mean.
Right. Yeah, the width of this ridge here, what would you say, 10, 15 meters? Kind of a little hard to judge, but...
Yeah, I think that's a pretty good estimate.
Yeah.
You know, it seems like it's not very far for the D2 to go from side to side,
Right.
and it's probably, what, about 5 meters wide?
Yeah.
Maybe not even that far.
A couple meters wide,
Maybe,
yeah. A couple, three.
Not more than 3, yep.
You know, in our maps here, we typically grid the sonar at maybe 50 meters at these depths. So we're not really even seeing. This is a little narrow feature, perhaps on a much wider feature.
Good point. So this is a rock pen, again, Ornithoptilum. I think this is now the 4th one that we've seen today. Very interesting to compare to the Helipterus whip-like sea pens that we saw earlier that were anchored in the rippled sediment. Another, I think, gastropod snail maybe there, and there's a young bamboo coral. And what's this? It must be a crinoid. So this is like that crinoid we got the close-up on. That it is a stalked crinoid, but it's got a lot of those long, cirri, feet-like structures coming off of the stalk underneath it. No, I think most of them are barnacles. They're kind of yellow, yeah. They're like acorn barnacles. You know, now we've been seeing a lot of these this whole dive, but I don't believe we've ever seen one with the feet extended. So
I don't think so. I've been looking for that.
Yeah, and if they were dead, you would expect that the plates in the center would open up, and so you would see a little hole in there. So I don't think they're dead, but maybe they work the night shift.
It's always a night shift down here.
Good point.
Well, there's our sediment.
Oh, wait, right there. There's one that's got its legs out.
Oh,
just in front of the hole.
Yep.
Yeah, above the left laser.
There we go.
It's not kicking the legs, but they're open. There we go. Confirmation that there is a little animal in there, and those are its legs, right legs and left legs. And they've got little setae that extend from them, so they kind of have, it's kind of like a net. It's kind of like they're moving a net through the water. Good stuff, Josh. Thank you. So earlier we heard from a couple of scientists on shore that they thought they'd also seen stalked barnacles or gooseneck barnacles, and so those wouldn't have the shell glued to the substrate. Instead, you'd see a little fleshy, what's called a peduncle, a muscular stalk, and then the plates would be above that muscular stalk. So I haven't noticed any of those since we first got to the bottom, but we'll continue to keep an eye out. So this whip looks like it has 1, 2, 3 branches, perhaps. So I need to come up with another name. Sparsely branching bamboo coral.
There you go.
How's that?
More adjectives. That
yes,
always helps.
More adjectives. Better. I think it's a worm, or no, it's a shrimp. You see a fish?
I think it's a shrimp.
Yeah.
Oh.
It's an amphipod. That is an amphipod. So what I'm trying to see, one of the ways you can tell the amphipods apart from the shrimp is that the shrimp would have 5 pairs of beating legs that it's swimming with, whereas the amphipod would have only 3. 2 of those legs would be modified into a sort of paddle-like structure. Naturally, it's swimming away, so it isn't giving us that opportunity. But amphipods are very, very common down here. It's a pretty good size one. Yep, I'm sticking with amphipod. When we look at the sponges, we often see amphipods sitting on the sponges, once in a while on the corals. But that's a good size one. There are many that are scavengers, and they will. There's different guilds. So one are compared to like vultures, so they'll stay several hundred meters above the water, above the bottom, trying to detect odor plumes from carcasses that have fallen to the bottom, and then they'll swim down and attack those. And then there's another guild that I thought are more like blowflies. They stay really close to the bottom, and they may even just sit in sediments until something dies nearby. Looks like there might be a brittle star hanging up in the crook of that arm. Isn't that interesting? Just dangling its legs into the water. Oh, this is good. Yeah. Oh, fantastic.
So
I wonder if Tim's still here. This is a different
I was
species.
gonna say, this looks different.
Absolutely. So these are fishermen for sure. We see these a lot. Interestingly, I've seen them on Halipterus sea pens like we saw earlier up in the Aleutians. You see them all the time on the sea pens there. But here's one in the bamboo coral. I've never seen one like this on a bamboo coral. And these guys will extend their arms out into the water column just like fishing rods, presumably to snag things that are floating by. And I should be able to get the name of this. I don't remember it offhand. That's very cool. This is definitely a new observation. And if anybody on shore knows the name, you can save me the time of searching. Please give us some insight. Well, there's a photo without an identification. Asteronix leveni. So I'm looking at some information from the folks at the Monterey Bay Research Aquarium Institute. They've done a lot of deep sea work out there, and they have a photo of one of these Halipter sea pens like we saw earlier with something that looks a lot like this. I'm not saying it's the same species, but it would be in the genus Asteronix. Thank you. Great. Good stuff.
Yeah, it's a great shot.
Yeah.
Wow.
We have got about 35 or 40 minutes of bottom time remaining. We are at a depth of 1,584 or 5 meters. Climbing this ridge-like feature that's extended southwestwards from the high point, which is 900 meters, and we're climbing towards a local high point, which can't be much more than, what are these contours, 10 meters?
Yeah. So I think around. I think 15 was our 1500 was our end of our line that we had drawn out.
60 meters or so more upwards, maybe 70. And when we started this dive, we saw the place we sat down was quite spectacular with lots of diversity of corals and sponges and various other organisms, urchins and crabs and so on. And then that kind of transitioned and changed a bit to where the corals weren't so dense. And was it then we started seeing crinoids, or did we see something else before that?
We
saw the sea pens.
Sea pens, right? We got into a poly, so we had a lot of sediment, and we could see the sea pens.
Right. And then we kind of moved out of those sediment pools, and then we got into this more, I don't want to call it flat rock. It's certainly not flat, but
definitely bare, yeah. So.
And the crinoids and the barnacles have really taken over, with the corals and sponges being much more spotty. So here again is another one of these crinoids that's apparently covered with these galls that are being caused by the presence, perhaps, of myxosomid polychaetes, Mysostome polychaetes. And there's also a couple of snails on there. So one thing I'd have to check, and maybe is Kevin. Yeah, Kevin, I see you're still in the chat room, so maybe you could tell us a little about the mysostomes. Are they feeding on the crinoid, or are they feeding on just taking advantage of stuff that the crinoid is capturing? I wonder if there's any data on that.
Well, some of it is Chuck Messing. Some of them, at least, are definitely parasites.
Oh, great. Okay.
Because some of them are found inside the gut of the crinoid. And of course, if they're forming galls, they're obviously irritating the animal. But whether they steal food or completely nibble on tissue, that's a question for Greg Rouse again.
Okay, thank you. We're getting a close-up now of another Chrysogorgia colony, and this may be a new one for the day, a new species for the day. You can see it's got very sparse branching, and maybe it's similar to the Metallogorgia in the respect that I don't see any polyps on the lower branches, so perhaps those will fall off later on. But you can see all the action is up at the tip. But this is definitely the genus Chrysogorgia. And you can see that golden-colored skeleton below a very, very thin tissue. Tissue so thin that the polyps almost appear to be balanced on the skeleton itself instead of embedded in tissue.
Well, a lot sparser than the ones we saw earlier.
Yep.
Yeah.
Yeah, right, exactly. So we were seeing the ones that had those squat lobsters in it. And we couldn't get a good view of the squat lobster because they were so hidden. And those polyps were thicker. The branches were more dense. Thank you. That's great. And no associates on that one, so no worms or lobsters or shrimp or... a brittle star, and a crinoid, another urchin. Plenty of echinoderms down here. So what's this? A snake? I guess it's a feeding trail, as I imagine what it is whatever sediments are on the substrate are being removed. There's a chitin. Are you kidding me? That's a chitin. Wow. So now we know what's making these trails. Fantastic. Oh, this is fantastic. So there's probably sediment and bacteria, and that's what the sort of grayish white stuff you're seeing. And then you can see where it's exposed, the black. And that's because this animal, which is a chitin, it's a type of mollusk.
Okay.
And if you go down to the seashore, these are another one of these organisms that's kind of streamlined to be able to survive crashing surf. And they basically have a mouth with a radula in it. The same thing that worm-like mollusk that was feeding on the coral I said before. And effectively it's a rasping tongue. And it scrapes the surface for, in this case it would be bacteria. In shallow water it would be plankton, phytoplankton or algae, or not phytoplankton because they're floating, but algae growing on the rock. So this is a deep sea chitin, and we can see evidence that it's been feeding in this area. This is a wonderful observation.
So he's polishing up the manganese crust here.
Exactly, he's getting it ready for you. When we're talking about it being shiny, it's because he's been doing all this work.
It's kind of like the Roomba of the deep sea here.
It's a lot like the Roomba of the deep sea. Good stuff. Good stuff. We don't see those too often. You can see, let's see, there's 1, 2, 3, 4, 5, 6, 7 plates, and the formal name for this group is Polyplacophora. And polyplac means multi-shell, many shell. So it's related to the snails. The snails have one shell. The clams and the oysters, those are called bivalves. The shell is divided into two, and it hinges in an articulation. These have 7 plates, sometimes maybe more or fewer. And they have what's referred to as a muscular girdle around those plates, and they kind of suction themselves to the bottom, and they especially do that in the crashing surf. Cool.
That's something new for us, this expedition. Great.
Absolutely. More of these, I guess, collapsed features. Nolan Barrett is sharing that they also saw a chitin on the last ROV leg, so that's great.
Cool.
Looks like the top of this bamboo coral isn't doing too well. Maybe another one of these fishing Asteronix ophiuroids. Geez, you know, as soon as you say something's rare, you never see it before. So, oh, good point. Oh, well done, Josh. So this is not the same ophiuroid. This is an ophiacantid. It's got those really spiny arms. So I don't think this is a fishing type. I don't know as much about the biology of these ones. None of above it. We have a crinoid, and I think there might have been a barnacle right up on the tip, and it might have been a gooseneck barnacle. Yep, several of them. 1, 2, 3, 4, 5 of them. And they have, you see that little short muscular peduncle just below where the shells are.
Right.
Most of these have their legs out. Great.
Yeah.
It's kind of broken up the surface here.
Yeah, we can see some sediment filling in now. It makes me think there's plenty coming down, just getting swept off the area where we were.
So when I see this, it's kind of smooth, but obviously this plate has kind of collapsed in place. So do you think that's something that's occurring very soon after the crust cooled, or is this something that takes place a long, long time after that?
Yeah, I mean, I think primarily these are probably, if these are tubes that are filling, they're probably collapsing relatively quickly.
Let's see. That is a primnoid coral. So now this is the third or fourth primnoid that we've seen today, and I'm not confident that that's the same one that we saw earlier. I think we saw 2 or 3 of the same species. I think this is a different species now. It's kind of interesting in a way. Again, it's tucked up under there. And this is not terribly uncommon for larvae. They may look for a place that's out of where the current is strongest, so they may tuck themselves out underneath, but then as an adult, they'll have to grow in a morphology such that they can extend the branches out to where there's enough current to feed. Thanks, Pilot.
Yeah, so sort of the growth shape of this ridge is certainly volcanic, but Deb Glickson in the chat room has been discussing this a little bit. The crust can kind of mask everything, and a lot of this texture you see is probably from the crust. It's tempting to interpret it as volcanic. In some places it looks like what you might expect if you saw a lava flow, but we don't know how thick the crust are. It's one of the things we'll find out when we get these rocks up. Again, it's the last dive, they're about 8 millimeters thick, and we're going north. We expect the crust to be a little bit thicker. Visually, they look a little thicker here, more of a mantle over the rocks overall.
So 8 millimeters, so that's the ones that we collected on dive 3?
Dive 3, yeah.
Okay, so that's definitely thicker than the ones from the dive prior to that.
Yeah, up on the Manahiki Plateau, that was a little further south. They were about 3 or 4 millimeters, and so then we went to 8, and then we'll see what these are.
Yeah, so it's interesting. One of the objectives of the Capstone overall project is to understand what are the diversity of life in the communities in these areas that are considered prime crustal zones because of manganese, and so that's why it's prime, right? The crust is getting thicker as we're going further north.
Getting thicker, and potentially the composition of it may be changing. That's one of the things we'll do with these samples as well.
Hey, Scott and Dale, this is Tim.
Hi, Tim.
I just got back. I was looking through the frame grabs of what I just missed in the last 15, 20 minutes, and I saw that you came across an Asteronix on a bamboo whip.
Yeah, exactly. I'm glad you said that. I was calling out to you at that time. Isn't that credible?
I'm sorry I missed it. It's amazing because normally we see them at the top of sea pens.
Exactly.
And the other coral we see them on is Radicipes.
Radicipes, good point. Right. So I'm seeing a theme here, Tim. You want to reveal what the theme is between Radicipes, the Halipterus, and that bamboo coral? They're all whips.
Yeah.
They're all whips. They're all unbranched.
Yeah. Yeah, you saw them on a branched bamboo whip, right?
Oh, well. So much for that theme. Thanks.
Yeah. That's all right. No, so that's really, really interesting. And it sort of looks like Leveni as a species. But
exactly.
The other thing is that you just came across a primnoid that had an ophiacanthid on it, and that's not the same ophiaroid that we've seen on the 2 previous primnoid.
And did we see that primnoid before? I thought that might be different from the ones we saw earlier.
That's a different primnoid and a different associate ophiuroid.
Great. Okay, thank you.
Saw.
So here's another one of my non-whip whips. So now I have to call these sparse branching bamboos. Got a new little branch coming out. So the interesting thing about this, and I've presented this at meetings, is it means you have to be careful about the identification because in some cases the names are associated with the fact that the colonies don't branch. Well, maybe they don't branch until they're 50 years old. All right? So you're fooled. And Astrid, I see, is back from her meeting. No, I don't know if I'm sorry to say this or I'm pleased to say this because you weren't there, but there have been no fish. And it looks like that amphipod again.
Right.
Yep. Here we have another of the crinoids with an ophiuroid brittle star climbing up the base, and there's one of the urchins. They're really hard to see from a distance, but when you get in close, it's more obvious that they're there. But that was the Plesiodiadema with the very long spines. Oh, he's decided he's leaving. Can't quite tell if that was voluntary or not.
That looked a little windblown.
Yep, I agree. This crinoid, Tim, also has one. Can we get a snap zoom to base of this new crinoid? So it looks like the same thing, Tim, and I'm noticing over on the far left there's another one that's got a brittle star on it. Huh. So I think this is two different brittle stars. The one that's on the stalk and then a different one that's on the base has a much rounder oral disc.
Yep, I agree.
Thank you. I think that's another one of the primnoids stuck down in there in that little cavity, and so perhaps the larvae are preferentially seeking out that sort of more benign current flow?
Well, you see, that's where the sediments are settling too, so that could also be where larvae are likely to settle.
Yep, that's what it is. That's primnoid. Thanks, Josh. That one does not have an ophiuroid associated with it. Brittle star. There we go. So,
what is that?
I'm trying to remember the name. No, it's a special kind of shrimp, not Procambrus. It's full of eggs, though, yeah. Or, excuse me, I won't say that it's full of eggs. I will say that there's a lot of gonadal material in there. It's not a polychelid, I don't think. It's too transparent. And I think they're more benthic. Tim, did you get a chance to see that swimming crustacean? There it is again. It's got some very large antennal scales.
It's not a polychelid.
Okay. Seems like a very inflated thorax.
Yeah.
Pretty big antennal scales. I'm not even seeing the legs.
Not sure. There's the pleopod. Yeah.
Huh.
Wow.
Yeah, indeed.
I don't know.
It's it's got all its thoracic legs just tucked up underneath it, and it's just swimming with those pleopods.
We got some great shots of it there, so someone can go back to it.
Nope. Well, that was pretty cooperative, actually. I'm impressed at the views we got of that. Go ahead, Nav, watch lead. 30 minutes, copy. Thank you. 30 minutes, folks. 30 minutes of bottom time. We are at 1,574 meters depth. It's 2.7 degrees Celsius, 3.2 milligrams per liter, of oxygen. We've been climbing this rather interesting feature and seeing the community shifting as we go. We obviously see a lot fewer sessile organisms up here. Been lots of echinoderms, though. Lots of crinoids, lots of urchins. Probably, I think I can count, maybe Diva, you can correct me, at least four different species of urchins, maybe even 5. It looks like we're coming up to
Agreed, Scott, at least 4 or 5.
Thank
I think
you.
there was like 1 or 2 Ctenopodina, and then there were like 3 Echinothuridae, the big pancakey ones. And then, is that it? Oh, and then the plesiodiadema, which we saw multiple times.
Right. Great. We've had good looks at this. I'd rather have a look at that yellow fan just in a little distance. Yep. Thank you. So this is likely another plexaurid coral. So plexaurid is the name of a family, the Plexauridae. It would be a member of the sea fans, the Gorgonian sea fans. And one thing we see fairly commonly, and we're definitely getting in the right depth zone, is a genus called Paramaricia. Now, it's hard to say. There are a lot of other genera, and they're very similar to each other, but often when we see them, there will be an astroscomatid ophiuroid with very long coiled arms on the colony. So we'll see if that's the case in this case as well. And sure enough, there it is. Tim, that looks like a pretty classic Paramaricia to me.
Yeah, and astroschema.
There we go. I said earlier we had a nice view like this. You could see the collarettes, which were the pattern of scleroids that are just below the tentacles, but above, you could call it, if you want, a neck. So you see there's the tentacles, there's a little bit of a white ring, and then there's a deeper yellow gold color. That's the soft part, the polyp body wall, and then it turns more white yellow below that. So that entire polyp can retract itself into that lower white part. And so we saw the Paragorgia earlier, and we saw the little slits, and people were wondering how could you feed. This is the same sort of thing. If we were to touch that with a manipulator, those polyps would all withdraw into that thicker part of the tissue, and it look much smoother.
So when you're out, you suddenly get a lot more surface area.
Exactly,
yeah.
exactly. Could see some of those barnacles. All three of them, I think, were open. I could see their legs were out.
Barnacles win the day by number of individuals.
Yeah.
That's for sure.
Well, yesterday I wanted to say the dominant species was the bamboo coral, but it had to be the crinoids living on the bamboo coral because there were often many of them.
Two or three on each, at least on each coral.
Oh, right. Good point. Nolan's reminding me and pointing out to Astrid that we did see something that looked like a gulper eel in the Sirius camera feed. So maybe you did miss a fish. Well, no, you did miss a fish. Question is whether it was actually a gulper eel. Sure looked like it. So another thing I'm wondering about when we're talking about the lack of suspension feeders here is not just the depth, but is the fact that we're following this ridge in more closely to sort of the massive part of this seamount. And so is that putting us more into a shadow, a current shadow?
Yeah, that could very well be.
There's a little colony up ahead in the center. And we have an Anthomastus on the left. Maybe another live snail. When I go hiking in the Chiricahuas, there's a place we go, Cave Creek Canyon. And as the name suggests, there's a lot of caves. And I'm always peeking in those caves, hoping there's going to be some interesting animal in there. And it's the same down here.
Yeah, I
want all these nooks and crannies to be full. Pilot, when we get in a position, can we get a close-up on the white colony that was just above off screen on the top of this flat surface? Understood. Wonder if that's a sponge up there. There's something different. We'll have a look at that later.
Might just be sediment. Looks like there's some pools.
Oh, is that what it is? I'm just looking at a bad angle.
I don't know. We'll have to get a better look in a second, I guess. But like off to the right, there's sort of
I see
those
that.
little pools there.
Let's see. This is another very small Primnoidae colony. So the family Primnoidae, it's an Octa coral. And what I used to be able to say that was the roughness of the polyps. I can see that there are scales on it, and the fact that the polyps are coming out in whorls or rings. You can especially see those scales, I think, on the back left. You can actually count them. And then that the polyps themselves are kind of pointed downwards. Fighting a little bit of current. There may be something else going on in that colony. I can't quite tell, but it looked like towards the top it was a little bit thicker in the main axis, right in there. Or maybe it's might be an ophiuroid hanging on. Maybe it's its arms that I'm seeing. Yeah. Yep, it's just an ophiuroid hanging around. So I think you can clearly see there are different plates that make up these polyps. They don't have a smooth body wall. That's a really nice image. Thank you, that's good. Let's move on. So I thought I saw sponge, but I think Dell is correct in pointing out maybe I was just looking at sediments and being fooled. And I guess I would suggest, given how little time we have, maybe we try to get as high up as we can just to see if the cap of this local high has more
I think that's a good strategy.
Yeah. Oh, it is a sponge.
Oh,
it is a
you're
sponge.
right.
Look at that, right above the lasers.
Okay.
Just when I said put on the gas.
It's a sponge on top of sediment, so there you go.
So we definitely haven't seen this sponge today. Shirley, are you still there? Do you want to comment on this? This looks like it might be a demo sponge. No.
Here, it's another hexactinella. Now, I wish Chris were here. I would kind of put this in like, but that's Faria.
Oh, Faria, yeah. I like that.
Chris is here.
Oh, Chris is there.
What is it?
Sorry, I just got down here. So I don't think the tubes are formed well enough to be in the family Furiidae, although there's some species in general like Spidoscopulia that don't have well-formed tubes. But my guess is that this is a uretid. It's in the family Uretidae. It's a hard sponge, so it's not so much hard, it's very brittle rather than soft and spongy. And this could be something that Henry Reiswig, working with a guy in Germany, Martin Dorman, identified as Heterorete in the genus Heterorete. It looks very similar to sort of this lettucey kind of a sponge that we have in Hawaii. So that would be my best guess. But really interesting. We don't see these very often, so it's an unusual sponge to come across.
Wow.
That was totally my second choice.
Sorry, guys.
Yeah, Furiid and Uretid are the ones that usually it boils down to.
I have a feeling, so we're looking at a homolid crab here, and it's one of these crabs we saw frequently on the last leg that likes to carry different kinds of cnidarians above it as a defense. This looks like it's carrying a little, maybe anemone, but I think it's caught a dandelion animal. It may have a rhodallid siphonophore in its arms. I've never seen anything feeding on a rhodallid siphonophore, so you may remember those. They look kind of like a floating ball with lots of weird little pulsations on it that have tentacles that act as anchoring lines. They go down to the rock. And boy, that sure looks to me like what's going on there. This crab has managed to capture one. Too bad. That was great. There were also, I noticed, some snails, little squat lobster, and some brittle stars on that sponge. Cool. Oh, cool observation. All right, so we're going to see if we can make a quick dash to try to achieve the local high if we can, and see if that local high is maybe out of a shadow enough to have a large density of suspension feeders up there as well. That was great. That was the first of that kind of sponge we've seen on this expedition.
Right. Yep.
Just in time, Chris walked back. It was Chris Kelly at the University of Hawaii. He was a sponge aficionado, especially with the glass sponges. I think that's a dead sponge. Well, it's a dead something. Didn't have to go too far out on a limb to get to that ID.
It looks like a very dead something.
Yep. Thanks for the clarification.
More adjectives, always better.
Always better.
Yeah, I think you're right. I think it used to be a sponge at some point in the past.
So, Chris, I don't know how much of the dive you're able to see out of the corner of your eye while you've been otherwise occupied today. I know you've been really busy, but it's been a really interesting transition. I know you were here at the beginning when we saw the sort of spectacular diversity of all kinds of stuff, really, but a lot of corals and sponges and urchins, other echinoderms. And then we got into an area where there were lots of pockets of sediment, and we had sea pens in those pockets. And then we moved out of that, and we've been seeing a lot of these barnacles. You can still see them yellow-brown dotting the surface here. These are all acorn-like barnacles. And then we started to see lots of the stocked crinoids. We eventually did collect one. We got a smaller individual, and lots of these crinoids appear to have myzostomid worms on them and the eulimid snails. And we've still been seeing occasionally a couple of different kinds of urchins, crinoids, both comatulid crinoids and stock crinoids, but a lot less of the sessile fauna. And we're wondering if maybe as we got a little bit higher and closer to the main edifice of the seamount that we were getting into a current shadow, or if it's just that we're changing depth. So we're hoping to reach the high, which is, about 15 meters up above us now?
Yeah, something like that.
Yep. To see if there's any change once we get up to the tip top. Again, not the top of the ridge, just the local high on the ridge, because after that it would go back down for a bit and then it would climb again up towards the main edifice.
Well, I didn't see the transition, but I did happen to look and see the collection of that crinoid, and I was just jumping up for joy when I saw that you grabbed that thing. You know, I could be mistaken, and I talked to Astrid about it when I just got down here, and she said that Chuck Messing was on the line advocating a collection.
Indeed.
And it looked to me like it only had four branching arms. Is that correct? Because I was trying to count the number of arms right before you grabbed it.
I have to admit that there's too much going on for me to even remember how many arms it had, but I can verify that Chuck was with us for a good hour to an hour and a half, providing us lots of information, and he was the one who directed us towards that particular specimen. We were looking for one that was a little bit smaller in hopes that maybe we could get the holdfast, but as you know, these things are usually in a position where it's almost impossible to get from the base. Oh, a fish. Stand by, Astrid. There it is. There is a little fish. There we go.
So Astrid is missing that she stepped outside because she hadn't had lunch.
Oh,
it's like 4 o'clock here.
That's why you don't go for lunch.
I'm going to see if I can bang on the window to get her to come in.
Looks to me like a midwater fish, Chris.
Yeah, I would agree. Boy, I don't know what that is. I really butchered my guess during that water column dive on that really beautiful fish you got. I'm sure you saw Bruce's email clarification on what that was. But yeah, I have no idea about this one. I guess is Bruce not watching right now either?
Yep, no, Bruce is unavailable. Amanda Netburn, who's here on the ship in the dry lab, agrees that this looks like a midwater fish. So we'll have to get Astrid to watch the replays, and it'll be funny to see her kick herself.
This is Chuck Messon. That looks like one of the stomiatoids. I'm not sure which one. It's a little outside my area, but I didn't notice whether that crinoid we collected had 4 arms or 5 either. I was so taken with the idea of collecting it.
Well, you guys don't have the
Yeah,
benefit of
Chuck,
having a video replay specialist next to you, so we're looking at an image. 1, 2, 3, 4. Chris, I think you're right. I think there's 4 arms that branch.
Yeah, I was studying it. I actually paused because I'd never seen anything like that crinoid, and I was studying it for a while. And I wonder if this is another one of the same ones that are coming up right now.
Yeah, I think so.
Can we get a quick zoom on this? 1, 2, 3, 4, 5. Well, this has got 5.
No.
So it figures that we collected an abnormal one.
Do you want us to put it back, Chuck?
No, I'll be happy.
Okay. Yeah, so when this, yeah, okay, so 1, 2.
Yeah, the other one's only got 4.
Yeah, it sure looks like it only has 4. But one of the arms then bifurcates twice immediately in succession. That's what's a little bit odd.
Yeah.
Huh. All right. Yeah, that's good.
Chuck, would you go to Hyacrinidae on this one?
As far as I know, the only Hyacrinus that's got any branching arms is Kahalewai in the Galapagos.
Yeah.
And so this one is the same as the big one.
That's a really good image there for us to.
If I can get a little better focus on the stalk, I don't think I've never seen a Hyacrinidae with branching arms like this. I think it's the distance between the branches is very different than the other crinoids I'm familiar with.
This was a good dive for you to tune into, Chuck. Been really blessed with the crinoids in this one. We got a purple holothurian. We've seen this now 7 or 8 times today, I think.
Yeah.
And I believe it's the only sea cucumber species that we've seen today. Chris, do you recognize that in terms of a genus or a family?
Well, I could take a guess on it. I think it's going to be a Synallactid. I think that's a reasonable guess, and it could be in the genus Hansenothuria based on the very small little tubes that are coming out of the back of it that have little black tips on them. That seems to be one of the characteristics of that genus. So that's my best guess, Hansenothuria.
Oh. Now this is interesting. Oh, this is great. I'm not sure if you know why this is so great. So we tried to scoop one of these up earlier today with the shovel. And the one we saw had only a single polyp. And we thought one of the reasons it was so exciting is there's only one species among the more than 2,000 species of described octocorals that grows as a single polyp. But here you can clearly see that one on the upper end has 2. So it was not what we thought it was. It may seem like I'm stalling. That's because I am, because I don't know what it is. I guess it's possibly something like an Anthomastus. I'm just not used to them growing in sediment. So it either means that there was an exposed hard thing under there that the polyp attached to and began growing, or it's something related to the Anthomastus, but it's not an Anthomastus. Chris, do you recognize this?
No, my guess would be Anthomastus, and like you, I assume maybe there's something underneath the sediment that it's sort of stuck to, but no idea.
Great, thank you. Good, good view. Good find. Thank you. We say Anthomastus, so we saw these pink ones that we were calling mushroom corals, and that's what we're relating that to. And one of the things we're doing is the relative size of the polyp to the whole stalk, but also you can see a lot of it look like warts. And I've discussed these a couple of times now. Those are presumably evidence that they have dimorphic polyps, meaning polyps of two different shapes. And one of them is very small, reduced, no tentacles, and is really for channeling water into the colony or for egg storage and so on.
You know, I haven't seen a barnacle in a while.
Oh, good point. And look, it's not smooth anymore.
Nope.
The substrate isn't smooth.
Yeah,
It's really...
It's definitely, they've favored that really flat, smooth surface.
Yeah, yeah, good observation. Looks like a couple of crinoids tucked in here. There's two stalked and a comatulid crinoid unstalked. So, Chuck, I don't know if you can see this, the small stalked one, the distance, I guess, let's see, 1, 2, 3, 5, it's also 5 arms branching, so maybe it's a smaller version of the larger one.
Yeah, I see this, I'm looking at this brown, the typical brown one in the crater, and then at the top is a much, much smaller one.
Right, you'll get a zoom in a second.
I see it now. Okay.
All right, I think we got to go. Hopefully you got enough of a view there, Chuck, coming up.
Could conceivably be a higher crinid, but it's so small that it's tough to tell. It certainly doesn't look like the arms are branching, although, that first big pinule might turn into an arm. There are a couple of crinoids that do that, although it's unusual. But that could be a higher crinid.
Nav's watch lead. 5 minutes, copy that, thank you. So we got our last 5 minutes to get a look at the bottom, folks, and then we'll be leaving bottom. As Del just noted, it's a lot rougher, really interesting. Why would that be, do you think?
Well, it could be we're on a little bit more of a slope there earlier, so maybe we're getting more sheet flows, so flat surfaces. And here it looks like the lava, when it came out, could have just broken up more, could have tumbled down a cliff or something like that. So we see these transitions on the seafloor, even in young lavas. A lot of it depends on the local slope and the viscosity and the and temperature of the lava when it comes out, and those things could vary quite a bit.
So the fact that we're on a local high on a ridge, could this be a source of all this?
Yeah,
lava?
so you can imagine these long ridges that come out. Those would have been places where a dike or a magma-filled crack would have propagated, and often along those dikes we see little local areas where lava sort of focuses and builds little mounds.
Oh, cool.
We see this all the time. We just saw something very similar in Axial Volcano when it erupted last year. We have these dikes going out from the magma chamber, and there are little spots along the top of the dike where we tend to get focused mound building, and this could be something similar to that, just probably an older example of it.
So we could, maybe just like be on the side of that little magma chamber. So Brisingid asteroid with its arms raised up in feeding position. No, I'd rather keep going up as much as we can in the last 5 minutes. Thanks. Yep. The sponge looks dead to me as we approach it.
Seen a lot of dead sponges today. As many dead as alive, I think.
Yeah.
So.
I did not mean that in the context that it may have sounded.
there may be some people out there that are disappointed by the fact that the community has gotten quite a bit sparser. But I find this really fascinating, these transitions between where you have very dense patches and then suddenly it all changes and then you get into a bunch of other things. And some of it could clearly be due to the substrate, as you pointed out, Scott, and some of these transitions could be due to oceanographic conditions that we're not aware of right now. But every time you see these transitions, they give you clues as to what's going on. It's just a matter of us being able to interpret what those clues are and understand what they mean. So I'm kind of actually really happy that you saw a bunch of different things going on today.
Yep, I absolutely agree with you, Chris. We're not going to learn how to interpret the pattern if we don't see variation along the way and then match that up to other conditions. So our last couple of minutes, I don't see that this is going to turn into a
coral forest again.
Coral forest, yeah, exactly. See the Anthomastus coral. I see an urchin in the background. There's that's interesting. Can we get a quick zoom on this ophiuroid right in the center right, bottom right, below the lasers? And I'm asking that because this sure looks from a distance like the kind that's normally got its arms wrapped around a coral, and you don't often see them just on the rock. So I'd like to at least get an image so that scientists on shore can have a look at that. Boy, that sure looks like an astroschematid ophiuroid to me. If Tim's on the line or Chris want to comment on this, maybe he's off in search of a new coral. Okay, that's good. Thank you. We got a good view of it.
See, my guess too, Scott. It sure looks like an astroschematid to me that should be on a coral.
Exactly.
So this guy hasn't found where it wants to squat yet.
Yeah, it looks like it's a pretty good size. He should have made a decision by now.
It's gonna have a long way to go here. I don't know.
Yeah, I agree with that. The only other time I've really seen something like this, sort of out in the open, was at the end of one of the dives from last leg. Very end of the dive, on top of a region kind of like this, had the astroschema was just spread out with nothing else around. So it's a little bit bizarre.
Well, if you believe in coincidences, it's the end of the dive again. So we've got that going for us.
I noticed, Chuck, also when you zoomed in on that ophiuroid, there was a lot of very fine organisms, just like a fine, almost an algal turf, waving in the wash of the ROV, and I wonder if those are agglutinating foraminiferans, because we've seen those branching. They look like little tiny trees on dead coral in Florida.
Yeah, Chuck, we've seen actually a lot of those arborescent foraminifera, and when we've collected some of the rocks earlier on this expedition, we've been able to preserve 5 or 6 of them. So they're definitely common down here, so it wouldn't be surprising at all to me that that's what we're seeing.
That's great. Thanks very much.
So I noticed right at the end there was a whip-like bamboo coral on the left side, and then tucked under the rock was another primnoid coral. So we did see a couple of corals at the end. I wouldn't exactly call this a forested cap.
Not quite a forest, but
must be above the tree line, I guess. So I believe, folks, that is the end of what was a pretty magnificent dive, a really interesting spot. Again, just to refresh your memory, this seamount was not mapped until last night. The mapping team did it, and so it was 5 a.m. this morning. We were all convening to find a spot to dive on, and we did. We are in the South Pacific, about 90 miles south of the equator, in the Pacific Remote Islands Marine National Monument in the Jarvis Island unit. So I guess it won't surprise anyone if I say that it was just mapped last night, so this is the first ever dive on the seamount. You wouldn't want to dive on it without a map.
No.
And so you have just explored with us and gotten the first glimpse into biological communities and the geology of this, and I really hope you enjoyed the dive. I think we had a good time doing it. And scientists on shore, give us 2, maybe 3 minutes just to get a drink of water, and we will start our pre-dive call to discuss what we're going to do tomorrow.
Right, and I'll just remind people out there that all of the data we've collected here today is publicly available. And so you can Google Okeanos Explorer, and you can get access to these videos, the bathymetry data, and lots of other stuff. And please continue to follow us.
And what's the depth here now when you're leaving the bottom?
We're leaving the bottom. We're like a 1535 right now.
Thank you very much.
All right, so as Scott said, we'll have a pre-dive call here for our next dive at Jarvis Island in just a couple minutes. We're just going to grab a quick drink, and we'll be right back.
I'll have to sign off and say goodnight, gentlemen, because it's late here